Hello students, welcome back to the microbiology course. Today we're going to discuss some of the most important bacteria. Now we've already discussed a few of the important bacteria, but we haven't discussed which group each of them belongs to, and that is something that a microbiologist needs to know. So you remember from first year biology that all of the living things on the world are organized they're all divided up into three different domains. There's the domain archaea, the domain bacteria, and the domain eukarya. Humans and plants and any organism that has a nucleus belongs to the domain eukarya. The archaea are primitive bacteria that were among the first life forms to exist on Earth, and they live in some of them are still around today, and so we'll need to learn a few of those. And then you have the domain bacteria, which are all of the bacteria. So you remember from biology 110 that all of these living things within the domain eukarya are divided up into smaller groups that are referred to as phyla or phylum singular. And the same is true for the domain bacteria. So the important bacteria are divided into similar groupings that have features in common that are phyla as well. So we're going to learn in order to be a proper microbiologist, we need to learn the important phyla that different bacteria belong to. Okay, so I'm going to discuss in this lecture, first we're going to begin by discussing different types of symbiotic relationships. This is how organisms live together in very close proximity. Bacteria living within the human body, for instance, they are symbionts, but we define the relationship between the bacteria and the human host differently depending on what the relationship is. So we will get into that. And then we're going to discuss obligate intracellular bacteria. You remember from the earlier lectures that an obligate anaerobe, the word obligate means obliged to or forced to live in certain conditions. Obligate aerobes are organisms that are obliged or forced to live in an atmosphere that has oxygen. Humans, for instance, are obligate aerobes. Obligate anaerobes are organisms that are obliged to live in the absence of oxygen. Oxygen will actually kill them, usually because they don't possess peroxidase enzymes that can detoxify hydrogen peroxide. That's usually the distinguishing feature one of the distinguishing features of obligate anaerobes is that they have no ability to detoxify hydrogen peroxide. So an obligate intracellular parasite or an obligate intracellular bacteria is a bacteria that can only exist inside a living cell, inside a living eukaryotic cell, for instance. So there are plenty of bacteria that can live on their own, but obligate intracellular bacteria are not among them. They have to exist inside a cell. So we discussed obligate intracellular bacteria a little bit when we talked about Koch's postulates. The fact that you can't grow certain types of bacteria in a bottle means that you can't confirm that they're causing a disease according to Koch's postulates. Okay, so we're going to discuss, there are a couple of examples of obligate intracellular bacteria. Most of the obligate intracellular parasites in the world are actually viruses, because viruses they're not actually considered to be a life form when they are outside of a cell. So viruses are the ultimate obligate intracellular parasites, but there are some bacteria that are obligate intracellular bacteria as well. Okay, so we'll discuss as we mentioned very early on in the course, all bacteria are generally divided into two categories, gram-negative and gram-positive. Right? And so among the important gram-negative phyla, the most important one is the phylum Proteobacteria. We're going to learn all about those, and there are quite a few of them. Then we're going to learn about the Spirochetes. The phylum Spirocheta is another phylum that contains a couple of the spirochetes that you already know about. Treponema pallidum is a spirochete, for instance. And then we'll discuss a couple of the cyanobacteria, which is yet another phylum. And all three of those phyla are composed of gram-negative bacteria. And then we're going to learn about two of the important gram-positive bacteria phyla. One is called the Actinobacteria and the other is called the Firmicutes. And so they are two phyla that are composed of gram-positive bacteria. Then we're going to learn about a type of bacteria which is actually we're going to learn about a grouping of bacteria that are referred to as the enterics. The enterics are members of the phyla Proteobacteria, so these are gram-negative. And they live in the gut of animals. They live in the intestine of animals. And so what happens is that they are frequent causes of sewage contamination of water or milk, and that's the only reason why we care about them. And so if you have enterics, these are members of a class within the proteobacteria called the class gamma proteobacteria. And if you take those enterics and it turns out that they can ferment lactose, we call them coliforms. And that's important because when you want to assess the purity and the drinkability of water or the purity and the suitability of milk for human consumption, you do a test called the coliform index. Right? So you take a sample of water or you take a sample of milk and you count the number of coliform bacteria that are inside that liquid, and then you refer to something called the coliform index, which tells you how many coliform bacteria can be present in that sample of water or milk before it's considered to be unfit for human consumption. So that's another test that microbiologists need to be familiar with, is the coliform index. Okay, and we mentioned the fact earlier on in the course that some bacteria have a GC content where 50% of their genome is made of GC pairs, so do humans for that matter. But in addition to that, there's some bacteria that have less than 50% GC content in their genome and others that have more than 50%, and that's yet another way that we can classify bacteria. And then finally, another way that we can classify bacteria is with a technique called the Lancefield grouping systems, Lancefield grouping system, which was named after a scientist named Lansing. Basically, she invented a method of grouping bacteria based on which antibodies they will react with, and we still use that technique today. It's called the Lancefield grouping system. It's a very useful and commonly used way to determine what type of bacteria is making somebody sick. If somebody has an infection, you do some tests and then you figure out which bacteria is infecting them based on which Lancefield grouping it falls into. And then at the end we'll have a couple of examples of archaebacteria. Okay, let me begin by explaining the different symbiotic relationships that microorganisms can have with other organisms, right? So symbiotic relationships are where two organisms live very close to one another or where one of them actually lives inside the other. So the different types of symbiotic relationships include neutralism. Neutralism is where two organisms live together. You know, if you have a bacteria living inside an animal, neither one of them really cares. Both organisms are unaffected. They're both sort of indifferent to each other's presence. That's called neutralism. Mutualism is the kind of relationship that E. coli has with human beings, for instance, because we all have 10 trillion E. coli cells in our colon, and we are providing them with food while they provide us with vitamin K. So that's an example of mutualism, where both of the organisms, where both parties benefit from this close relationship. They both benefit from this symbiotic relationship. Amensalism is where one organism is affected and the unaffected and the other is actually harmed. So usually you have some bacteria that's living in an animal or a human, and it doesn't care whether it's living inside the animal or not, but the animal cares very much whether it is or not because the animal is actually damaged by the relationship. Okay, now commensalism is where one organism benefits and the other one is unaffected. So there are many bacteria that live inside humans or other animals that benefit while the human doesn't really care, doesn't benefit from the relationship, but is not at the same time is not harmed by it. Right? And then finally, parasitism. Parasitism is where you have a parasite living inside you. And a parasite is where the parasitism is where one of the organisms, usually the bacteria, benefits at the expense of the other. So the other is harmed. Right? So many of these bacteria that are pathological, pathogenic bacteria are usually parasites. So they get inside us and they damage our tissues, and they start eating us whether we're dead or not. And they gain a lot from doing that, but we don't gain anything. In fact, we are harmed by that relationship. So most or all of the pathogenic bacteria that we discuss in this course are in fact practicing parasitism. They are therefore parasites. But I mentioned in the first lecture that there is a subdiscipline of microbiology called parasitism that refers to protists and helminths infecting people. Right? So protists and helminths infecting people. Okay, so don't confuse the two terms. Parasitism is it's a general term that biologists use to describe this type of a relationship. And parasitology usually refers to a branch of microbiology where people just study protists that infect people, like the malaria, like Plasmodium vivax, for instance. All right, so some bacteria are facultative intracellular bacteria, which means they can live either inside or outside of a eukaryotic host cell. But other bacteria are what are called obligate intracellular bacteria, which means they can only live inside eukaryotic host cells. Not all of them are pathogenic, but those that are, this being an obligate intracellular parasite is actually quite handy. It's quite fortunate because you can evade the human immune system. Remember that the human immune system is mostly white blood cells and the phagocytes, macrophages, and so on. And they cannot, if you run and hide inside a human cell, those white blood cells cannot pursue you into that cell. They're stuck on the outside. So you've essentially gained a sanctuary by escaping inside these cells. You've gained yourself a sanctuary. All right, so as I said, viruses are exclusively all viruses are obligate intracellular parasites, but there are only a few bacteria that are. And two examples that we're going to learn that are obligate intracellular parasites are any members of the genus Chlamydia and any members of the genus Rickettsia. And these two geni are both pathogenic. So a little later on, we're going to discuss Chlamydia trachomatis, which is a sexually transmitted disease, and also a bacteria called Rickettsia rickettsii, which causes an illness called rickettsial fever, or Rocky Mountain spotted fever is a common name for it. We have both of these things endemic to BC, to British Columbia. People in British Columbia catch chlamydia by having the wrong sexual partner and so on. And people who go out camping in the Rocky Mountains will sometimes get Rocky Mountain spotted fever, which is caused by a bacteria called Rickettsia rickettsii, which is carried by a tick vector. It's carried by a tick vector. Okay, so Chlamydia and Rickettsia are two examples of obligate intracellular bacteria. You should remember that. You should memorize that. All right, now let's go on into our discussion of important bacterial phyla. So all living organisms are grouped into domains, the three domains: bacteria, eukarya, and archaea. And then within the domain bacteria, we have several phyla. Right? So there are a very large number of bacterial phyla that are composed of gram-negative bacteria. Right? So the largest phylum of all, that contains by far the most bacteria, is the phylum Proteobacteria. Right? This is the largest phylum, and all of the bacteria within the phylum Proteobacteria are gram-negative. Next, the next phylum is the Spirochetes, right? So they are also gram-negative. And then finally the Cyanobacteria, and there are a few others that we're not going to bother with. Right? Now the interesting thing about the Cyanobacteria is that they are gram-negative, yes, but they are also photosynthetic. They are photosynthetic, so they can generate energy from sunlight, and they are green in color. Most of them are green. Some of them are red, but most of them are green in color. And we believe that cyanobacteria, certain forms of cyanobacteria, were the original chloroplasts. They are the origin of chloroplasts in plants. Right? So at some point, a eukaryotic cell, at some point 2 billion years ago, or not 2 billion, but probably 1 billion years ago or less, some eukaryotic cell swallowed a cyanobacteria, and instead of digesting it, it just kept it, and then that cyanobacteria divided and multiplied inside that eukaryotic cell to become what we now call a chloroplast. All right, now let's look at the gram-positive phyla. There are two main ones. Actinobacteria. The actinobacteria are gram-positive and they have a high GC content, and there are several important ones that are important to humans. And the Firmicutes they are gram-positive and they have a low GC content. Right, so we'll discuss those phyla as well. So let's start with the important gram-negative phyla. Starting with the largest one, the phylum Proteobacteria. Again, they are all gram-negative. There are 5 major classes of proteobacteria. So as you remember from phylogeny, when you studied phylogeny in first year, you remember that you classify things, everything belongs to a domain, and then you classify it according to a phylum. And then within a phylum, you can group things according to what's known as classes. So the next level of organization are the classes. And there are 5 major classes within the phylum Proteobacteria, and they are all named after the first 5 letters in the Greek alphabet. The first 5 letters in the Greek alphabet are alpha, beta, gamma, delta, epsilon. Alpha, beta, gamma, delta, epsilon. And so the classes are alpha Proteobacteria, beta Proteobacteria, gamma Proteobacteria, delta Proteobacteria, epsilon Proteobacteria. And so you should memorize what the 5 classes are within the phylum Proteobacteria. So let's start at the beginning with the alpha Proteobacteria. Okay, so here are some of the important bacteria that you will find inside the class alpha Proteobacteria. The 2 that I've colored in green at the top are important to agriculture. Right, so if any of you have any relatives that are in farming or agriculture or anything like that, these 2 bacteria will be important to you. So Rhizobacterium is a priceless, irreplaceable bacteria that is involved. The Rhizobacterium is the genus name, right? So there are several species within this genus. And they are involved in nitrogen fixation, which means that where do we get the nitrogen to make our amino acids? Well, obviously it comes from nitrogen gas in the atmosphere, but how on earth do you convert nitrogen gas in the air into ammonia, for instance, that can then be converted into amino acids? That can be done by only certain organisms. Most plants can't do that. Most plants are not able to do that. They can only do it because they have rhizobium bacteria living inside their root cells, inside their roots, right? And so there's a certain type of a plant called a legume, right? Legumes are a kind of a bean-like vegetable. You can look up legumes. Certain plants are classified as legumes, and they make these kind of bean-like structures. And so the legumes have roots, and within the roots they have little bulges called nodules, root nodules. And within the bulges, within the root nodules, you will find rhizobium bacteria. Okay, now moving on. Agrobacterium is another genus of bacteria, gram-negative bacteria, that's important to the agriculture industry. And in this case, it's not beneficial. It's a pathogen, right? So the members of the Agrobacterium genus are plant pathogens that produce tumors, which are commonly called crown gall tumors. Right? So these plant pathogens produce ugly, unsightly bulges on plants that make them unusable. Right? So those are the Agrobacterium. Now, the fact that Agrobacterium are important to agriculture might lead you to believe that they're beneficial just by the name, but it's a little counterintuitive. Counterintuitive means the opposite of what you might think. So the agrobacterium has the word agriculture, the root word for agriculture in the genus name, but they are not beneficial to agriculture. They are actually harmful to agriculture. All right, next genus, Brucella is a genus of bacteria. There are many species within that genus, and it is a common human pathogen that's found in cow's milk, right? And it causes brucellosis. Brucellosis is a fever, right? So brucellosis is a fever. It's accompanied by a high temperature. And it is a common contaminant of cow's milk, which is the reason why we pasteurize cow's milk. Pasteurization means you heat it up to a temperature that will kill the bacteria that are in it hopefully without damaging the milk too much. Right? So you know that milk is a little tricky. If you heat it too much, it will coagulate, right? You'll definitely spoil it. So the trick is you have to heat it just enough to kill the bacteria, and if you heat it so you have to find the right temperature which will kill the bacteria that are contaminating the milk without actually spoiling the properties of the milk. So that's what pasteurization is. All right, now the next genus that's of interest to us is the genus Chlamydia. Genus Chlamydia. This, as I said earlier, is an obligate intracellular pathogen, and in humans we care about it because it's a human sexually transmitted disease, an STD. STD stands for sexually transmitted disease. It's something that humans get, if they pass around to each other during sexual activity. Chlamydia. Right. Rickettsia is another genus of bacteria that is carried by a tick vector, and it causes an infectious disease called Rocky Mountain spotted fever. Infectious, but humans don't transmit it directly to each other. It's transmitted indirectly through a vector, tick vector, an arachnid vector. Now, one of the other interesting things about Rickettsia is that you have to memorize is that Rickettsia is an example of a pleomorphic bacteria. In the last section, we discussed what the term pleomorphic means. It means it has no consistent shape. Right, so pleomorphic has no consistent shape means I could very well ask you on a test, give me an example of a pleomorphic bacteria, and you would say Rickettsia or Rickettsia rickettsii. Okay, so starting over again. Here we have a root of a legume plant, right? And it has these little bulges that are called root nodules. And within the root nodule, you have many different rhizobium species. Right? Here's another picture. On the left, we have an actual photograph of root nodules. And then on the right, somebody cuts open the nodules, and you see a lot of bacteria inside. So the example which I might ask you about as a multiple choice question, Rhizobium leguminosarum contains the word legume, right? And so that's a dead giveaway that this is the bacteria that is advantageous to legume plants, right? And they are involved in nitrogen fixation. They allow the plant they take nitrogen gas out of the air and they convert it into amino acids, and then the plants get to take those amino acids, right? So they are involved in nitrogen fixation. Without nitrogen-fixing bacteria, as they're called, without nitrogen-fixing bacteria, humans would have only a very limited number of plants to eat. Humans and other animals would have probably only 10% as many plants to eat as we do now. So we owe a great debt of thanks to the nitrogen fixation bacteria. All right, as I said, agrobacterium sounds like it should benefit plants, agriculture, but in fact it's the opposite. It creates these hideous, ugly, bulging structures, tumors, plant tumors, and they spoil crops. So you see here this particular bacteria is creating an ugly, hideous structures, tumors on corn, so that you can't sell that corn or eat it. Okay, here is a plant with something called a crown gall tumor, and that's another common deformity of plants, which is caused by Agrobacterium species infecting the plant. So another multiple choice species that I might ask you about is Agrobacterium tumefaciens. Tumor contains the word tumor, so you know what a tumor is. It's that bulgy, non-differentiated structure. And faciens is the Latin root word for face, so it means a tumor on the face of the plant. All right, Brucella causes brucellosis. It's a bacteria that is a common contaminant of milk. On the right, we have a cow being milked. People generally do not drink the milk directly as it comes out of the cow because they might get brucellosis. Brucellosis is a fever, right? So therefore, brucellosis as a disease is classified. Brucellosis as a disease is classified as a zoonotic infection. It's classified as a zoonotic disease because you catch it from animal products. This problem was solved by a famous French scientist named Louis Pasteur, who's shown on the left. And the technique that he invented was to heat up the milk just enough to kill the bacteria, particularly the members of the genus Brucellosis, and that's called pasteurization. So two examples of Brucella genus are Brucella abortus and Brucella brucei. So you should memorize those two, that they are two common contaminants of milk. All right, chlamydia. As we know, chlamydia is a sexually transmitted disease. It's spread through sexual intercourse, and the members of the chlamydia chlamydia genus are obligate intracellular parasites. They cannot grow by themselves. If you put them in a bottle with some water and some sugar, they will not grow. They have to be grown inside. In the lab, we use things called layers of feeder cells. So you grow some animal cells in a bottle, and then the chlamydia will live inside the animal cells. Okay, so memorize the name Chlamydia trachomatis. Chlamydia trachomatis is the cause of sexually transmitted diseases, asexually transmitted disease. Now, when chlamydia reproduces, what it does is it gets into a cell, and then it creates living cells that are called reticulate bodies when you see them under the microscope. Reticulate bodies. Right? You end up having enough of them that the cells that are infected produce something called an inclusion. Right? So inclusion, or commonly known as inclusion bodies, means things that you find inside the cell. Right? So the term inclusion or inclusion bodies is a fancy sounding word that simply means things small objects that you can see inside cells, inclusion bodies. Right. However, if you're a microbiologist, you'll hear the term inclusion bodies all the time, so you should be familiar with that word, that term. Right? So they form an inclusion body, and then some of them kind of form sort of something like a spore, and those are called elementary bodies. And these are not living things; they're sort of basically a spore. And then the elementary bodies burst out of the cell and infect new cells, usually epithelial cells. Right? And then they get inside and they form new reticulate bodies, which lead to new inclusion bodies, which lead to new elementary bodies, which come out and infect other cells. And that is the life cycle of chlamydia. Chlamydia trachomatis, for example. Okay, Rickettsia. Rickettsia causes Rocky Mountain spotted fever and has a tick vector. Okay, so there's a tick. You can see that's a human finger. You can see how small the tick is. They are very difficult to see, but you have to be vigilant. And if you're out in the wilderness in British Columbia, you have to look out for these, because we do have these in British Columbia, particularly in the spring and the summer. Not so much in the city, but in the woods, right, where the primary host, or the main host for ticks is fauna like deer and rabbits and things like that are full of ticks generally. And they carry Rickettsia, among other things. They also carry Lyme disease, Borrelia burgdorferi. And we have both of those things in British Columbia. So both of those things are endemic to British Columbia. Okay, so Rickettsia is an obligate intracellular parasite, and it causes a disease called Rocky Mountain spotted fever, which is also known as a rickettsial fever. Rickettsial fever. And the other thing that you need to memorize about the genus Rickettsia is that it is an example of a pleomorphic bacteria. So if you look at them under the microscope, the cells have no fixed shape, and you won't see them under the microscope unless you got them out of a human cell or an animal cell. Okay, so there's how big the tick gets after it's finished sucking your blood. It's big enough to see at that point, or big enough to see more easily. And by the time you see it, it's too late, right? It's already injected you with Rickettsia, for instance. Okay, so there is a big tick. They like to burrow under the skin and suck the blood out. One of the common places to find them is under your hair. So if you're out camping, it's important to comb your hair and keep your hair. You comb your hair frequently with a fine-toothed comb to make sure that no ticks have snuck in there and they're not able to bite you. Another thing is if you're camping in a tent or something like that, you have to you take your boots off at night so you can sleep in a sleeping bag. You need to check your sleeping bag for the presence of ticks, and then when you put your boots back on in the morning, you need to dump out your boots, shake out your boots to make sure that no insects or arachnids got into your boots while you were asleep. So and then you also have to check periodically to make sure that you don't have any ticks underneath your clothes. So here's a picture showing the ticks inside cells, inside living cells. Okay, so it causes a rickettsial fever, also known as rickettsia, and the example species that I need you to memorize is Rickettsia rickettsii. A little footnote down here at the bottom, Rickettsia does not cause rickets. Rickets is a bone disease that's caused by a lack of calcium in the diet or a lack of vitamin D used to absorb the calcium. So it has nothing to do with that. The name is just similar, so just be careful not to mix up the names. And one of the signs of rickettsial fever, so one of the signs of rickettsia is that you have petechia. You have either petechia or purpura. You have a petechial rash or a purpural rash. And this is caused by capillaries under the skin bursting to cause red spots. Hence the name spotted fever. So you have a fever, so you feel hot, and you break out in these spots. The only difference between petechia and purpura is that petechia are small and purpura are large. So purpura are 1 centimeter in diameter. Petechia are, a third of a centimeter. So I might ask you a multiple choice question about that. So I say, if you have red spots, you're covered in a rash of red spots, and those red spots are 1 centimeter or larger. Do you call it a petechial rash or a purpural rash? And the answer is, in this case, a purpural rash. There are a lot of other diseases that give you a petechial rash. Okay, let's move on to class beta proteobacteria. Okay, so 3 that we care about. Leptothrix is a relatively harmless aquatic bacteria. The word aquatic means it lives in the water, right? The word aquatic means that it lives in the water. And it has the ability to oxidize iron in order to live on it. So it can actually rust. It causes rust in pipes, for instance. Right, so if you have a water pipe that becomes all rusty, it may be because the air reacted with the metal, but it could also simply be because Leptothrix bacteria caused the pipe to rust. All right, next is our old friend Bordetella, genus Bordetella, example Bordetella pertussis, which causes whooping cough, easily spread by droplets, highly contagious. It's a respiratory disease that is generally spread through children at daycare centers and things like that, children coughing on each other or sneezing on each other. Children are notoriously bad for spreading disease because they are notoriously unhygienic. They wipe their nose and then they touch your face with their hands, or they touch each other's faces all the time. That's something that children do, but adults don't do that. Generally, that's an easy way to spread sicknesses. And so you have to be particularly careful with children when they're playing with each other, because they have a lot of unhygienic habits, which we have to train them out of. But it takes a little while, of course. Okay, and then of course we have the genus Neisseria. Neisseria is also a member of the beta proteobacteria. Our old friend Neisseria gonorrhea is a sexually transmitted disease that causes a sexually transmitted disease called gonorrhea, but it can also cause meningitis depending on where the bacteria goes. If the Neisseria is spread, well, I won't go into detail, but if the bacteria is spread through sexual contact, it depends on where the different body parts are. It is in fact possible for Neisseria gonorrhea to be spread from the genitals of one person, from the reproductive organs of one person, to the eyes or the ears of somebody else. And therefore, that from the eyes or the ears, it's fairly easy to get into the brain. And when the brain becomes inflamed, or the membranes around the brain, of course, are called the meninges. If the meninges around the brain are inflamed, then you have meningitis, which is potentially lethal. So Neisseria gonorrhea is uncomfortable and unsightly, but it's usually not deadly. However, meningitis can be deadly sometimes. All right, so starting from the top, Leptothrix can cause rusting pipes. It's an aquatic microbe, member of beta proteobacteria, class beta proteobacteria, can cause rusting of underwater pipes. Bordetella pertussis. Bordetella pertussis causes whooping cough in children. There is actually a vaccine to prevent it, which children can get, and adults can get it as well. Neisseria gonorrhea, sexually transmitted disease. A few more words about Neisseria gonorrhea. So Neisseria gonorrhea, you will remember from the lecture on morphology that Neisseria gonorrhea is a diplococcus, and since all of the proteobacteria are gram-negative, it is a gram-negative diplococcus. Neisseria gonorrhea is the only negative diplococcus that I'll teach you about in this course, and so if I ask you for an example of a negative diplococcus, you can tell me Neisseria gonorrhea. Okay, they are members of the phylum Proteobacteria and class Beta Proteobacteria. All right, they are spread often by sexual contact, skin-to-skin sexual contact. They are not generally spread through droplet transmission or fomite transmission. They can be transferred, it's not a transplacental infection, but they are transferred from mother to child during childbirth. So if the baby's head comes out through the vagina, and the vagina is infected with Neisseria gonorrhea, the baby could die from meningitis, because the Neisseria gonorrhea will get into the baby's eyes on the way out through the birth canal. And so from the eyes, it's a relatively short trip from the eyes or the ears into the brain, cause inflammation of the meninges, and it is possible for the baby to die from meningitis. So for about a century, we've known this, more than a century. And so the common practice is that when the baby comes out of for most of that century or so, when a baby is born, the doctor will rinse the baby's eyes with a 1% silver nitrate solution. When we get to lecture 11, when we get to lecture, I think it's lecture 12, sorry. When we get to lecture 12, we're going to find out that metal ions, for various reasons, metal ions can kill bacteria. And so one of the common ways to kill bacteria on surfaces is to take a solution of metal ions and kind of wash the surface with metal ions. So for 100 years or so, whenever a baby was born, the doctor would bathe the baby's eyes in a 1% silver nitrate solution in order to kill any Neisseria gonorrhea in case the mother has gonorrhea. It's not, depending on how far along the case is, it may not be easy to tell. The mother may not, it may be a subclinical infection, so the mother's unaware that she has gonorrhea. And so just in case, they would just rinse the baby's eyes with silver nitrate. If you're seriously worried about it, you can take the baby out another way, which is called a cesarean section or a C-section. This is where you do surgery and you cut the baby out so that the baby, you cut open the mother's womb and then you take the baby out that way so that it doesn't go out through the vagina. That, of course, is surgery, which is quite dangerous. So they generally, obstetricians do anything they can to avoid doing a cesarean section. Okay, and finally, Neisseria gonorrhea is the only bacteria that I know of anyway that does a little bit of antigenic variation. So it has proteins on its surface and it has several genes that encode those proteins, and it can turn every other generation will turn on or turn off a different set of those genes to stay one step ahead of the immune system. So Neisseria gonorrhea is very difficult to get rid of using regular human immune system. The best way to get rid of it is with antibiotics, and I explained earlier in the course that we're rapidly running out of effective antibiotics because we are abusing the use of antibiotics by prescribing them to people who don't need them, or people who have diseases that are not affected by antibiotics, like viral diseases, for instance. All right, now depending on so generally Neisseria gonorrhea is spread through sexual activity, but depending on what type of sexual activity, and I won't explain them, but you can guess, you can imagine, there are several different types of sexual activities, and depending on where the bacteria touches the other partner, the other person, you could end up with any of these diseases shown on the right. So urethritis, of course, is an inflammation of the urethra. The urethra is where the urine comes out, and we learned earlier on in the course that Neisseria gonorrhea is virulent because it has structures that adhere to urethral epithelial cells. Okay, now proctitis, procto refers to the rectum, right? So that's an infected rectum. Pharyngitis is an infected throat or mouth, right? So that's another type of sexual activity entirely. Cervicitis comes from regular sexual activity. The cervix, of course, is the ring of muscle that divides the vagina from the uterus. It guards the entry to the uterus, and so if it gets infected with Neisseria gonorrhea, it can become inflamed. And finally, if you have the ovaries, the fallopian tubes, the urethra, the vagina, and the uterus, all of those structures are inflamed due to gonorrhea. That's referred to as pelvic inflammatory disease, PID. Right, so you do need to know that. I might ask you what PID means, and I might ask you what it is, and I might ask you what causes it. So here we have pelvic inflammatory disease. It's basically where the entire female reproductive system becomes inflamed due to an infection of Neisseria gonorrhea. That's pelvic inflammatory disease. All right, let's move on to class gamma proteobacteria, including the enterics. Okay, so here are several of them, right? So up there at the top is our old friend Escherichia coli. Next, there's a genus called the Enterobacter genus, example Enterobacter cloacae. The reason I mentioned that one is because it has been implicated as a possible culprit in causing obesity. Right, so people who are obese have excess weight, which they don't want to have. They're heavy and have adipose tissue, fat tissue, to the point where their life is actually endangered by it. Right, and their life and their health is endangered by being too big and too heavy and having too much adipose tissue. Right, now remember that we shouldn't be judgmental about people who are obese, because a lot of it has to do with your metabolism. And it turns out that, as well as you know, just by looking at men and women, you know that men and women put adipose tissue on their body in different places, which is proof that the adipose tissue being deposited on your body is a genetic thing. It's not a matter of how much you eat necessarily. And some people have a very efficient digestive system that extracts very much energy out of a little bit of food. That used to be an advantage, because when humans were hunter-gatherers and they didn't have regular access to food, it was a definite survival advantage to have a metabolism that could do that. Right? So suddenly now, so that for 200 years of human existence, it was an advantage to have a metabolic enzymes that can extract a lot of energy out of your food. And then suddenly, in the last 100 or 200 years, we decided we don't like that anymore, and we're starving ourselves to get rid of the adipose tissue. That's an interesting problem. You can talk to the anthropology and sociology teachers about that. But from my perspective, as the microbiology teacher, I'm just pointing out the fact that obesity may be caused in part by various members of the Enterobacter genus. Okay, next, Klebsiella. Example, Klebsiella pneumonia. That is an enteric bacteria that lives in the intestine, but as the name implies, pneumonia, anywhere you see the word pneumo, probably has something to do with air or breathing. So Klebsiella pneumonia can also occasionally cause pneumonia in the lungs. If it somehow manages to jump from the intestines to the lungs, sometimes it does that through the oral fecal route. Okay, Salmonella, genus Salmonella, example Salmonella typhi. Salmonella typhi was famous for causing typhoid fever, which caused many epidemics in Europe, killed millions of people over the ages. Yersinia, genus Yersinia is also classified as an enteric. If it gets into the wrong place, if it manages to get its way into the lymphatic system, remember the lymph is the parallel network of tubes that run around the body, and the lymphatic system has lymph nodes. If Yersinia manages to get from the intestine into your lymphatic system, it will cause the bubonic plague. Bubonic plague killed half of Europe in the Middle Ages, and it was believed to be carried by rats, but it was actually carried by fleas, by a flea vector on the rats. So Yersinia pestis was the cause of the bubonic plague, which killed half of Europe in the Middle Ages. It still exists today. Okay, next, Shigella. Example, Shigella dysenteriae, causes dysentery. Dysentery is violent bloody diarrhea, which causes extreme hydration and blood loss. So it's a very bad disease to have, and dysentery is caused by a number of different microbes, only one of which is bacteria. So Shigella dysenteria is one of the bacteria that can cause dysentery. Entamoeba histolytica is a protist that we'll talk about in a week or so that causes dysentery as well. Okay, underneath that we have Legionella, genus Legionella. Legionella pneumophila is also known as Legionnaire's disease. Any of you that speak French know that a legionnaire is the French word for soldier. And in North America, the United States and Canada, the word legionnaire is not applied to any soldier. It applies to a retired soldier, right? So if you look around Vancouver, you'll see all of these bars, these drinking establishments that are called legions, and what those are clubs for retired soldiers. And they became very popular in in many places. They became popular after World War I and World War II, when there were literally millions of legionnaires around who'd just been discharged from the armies. The armies were very big during the war. Most of them have died out by now, and there are not that many people in the army anymore. But those that are around still, when they retire, they can go to the Legion. And so they go to the Legion and they drink and they pal around with each other and they socialize. And they sometimes have conventions. And at one time in 1970, there was a Legionnaires' convention in the United States where a particular type of bacteria infected a whole bunch of these old men who were retired soldiers and killed a bunch of them. And for that reason, it was named Legionnaire's disease. And the bacteria, which had been unknown, was named Legionella. And the word pneumophilia means it likes the lungs, right? So this is a lung disease. It's typically spread in air conditioners, and so we'll talk about that. Okay, now below that we have Pseudomonas, genus Pseudomonas. Example, Pseudomonas aeruginosa. you should remember Pseudomonas aeruginosa because it is a critical type of bacteria, a nasty type of bacteria that often infects wounds. It infects surgical incisions and wounds, but surgical incisions. So if you go to the hospital to have your appendix taken out and then you get this disgusting green slime growing on the incision that they've sewed up, that means that they haven't been changing the dressings properly and some dirt has gotten underneath the bandages. And if it's green, if it's a fluorescent green slimy color, that is probably Pseudomonas aeruginosa. Now, the reason why this is critical that you know about Pseudomonas aeruginosa is that you will then learn something that apparently a lot of people who are involved in cleaning the hospitals do not know. I mentioned to you before that what do you remember what you call an infection that you acquire while you're in the hospital? You call that a nosocomial infection. One of the common nosocomial infections, nosocomial infections is caused by Pseudomonas aeruginosa. It shouldn't be in the hospital, and the reason why it is because some hospital managers are unaware of the fact that Pseudomonas aeruginosa actually likes many of the solutions that you use to clean hospitals. Right? So if you want to clean a hospital and get rid of the Pseudomonas aeruginosa, you would use diluted bleach. If you want to encourage the growth of Pseudomonas aeruginosa, you would use a lot of the common cleaning agents that you buy in the store that are listed as antimicrobial soaps. Right? So antimicrobial soaps are soaps that contain different compounds, chemical compounds, which we'll talk about later, that kill most bacteria with the exception of Pseudomonas aeruginosa, which actually likes them. Right? So you're encouraging the growth of Pseudomonas aeruginosa if you clean your hospital with some of these triclosan derivatives and things like that. All right, now way down at the bottom we have Vibrio cholera. We learned about that at the beginning of the course, or we certainly learned about it during the morphology lecture because it is a Vibrio. In fact, so you know that Vibrio cholera is Vibrio shaped. How many flagella does it have? Do you remember? Because you do need to know this. All right. It has one, so it is a monotricious Vibrio. And now you know that it's gram-negative because it's a member of the proteobacteria, and all members of the proteobacteria are gram-negative. And so Vibrio cholera causes a waterborne disease called cholera, which causes diarrhea. The symptoms are diarrhea. So people who die from cholera usually die from dehydration. That's a result of the diarrhea caused by cholera. It is spread through the oral fecal route. So you have drinking water that has been contaminated by sewage, and some of that sewage came from people who already had Vibrio cholera infection. All right, these genus Escherichia, genus Enterobacter, genus Klebsiella, genus Salmonella, genus Yersinia, and genus Shigella are the enterics. They are classified as the enterics. And so I could very well ask you a multiple choice question, say which one of these is a member of the enteric family, which one, and I'll give you like three bacteria that are not and one that is, and you have to pick that one. All right, so we have the phylum Proteobacteria, the class Gamma Proteobacteria, and within the class Gamma Proteobacteria we have the order Enterobacterales, and within the order Enterobacterales we have the family Enterobacteraceae. The family Enterobacteraceae are the enterics. These are the enterics. These are all members of the family Enterobacteraceae. These three, specifically Enterobacter, Escherichia, and Klebsiella, these three ferment lactose. So how would you tell? How would you tell if they ferment lactose or not? What's the easiest way to tell? What would you plate them on? You would take a sample of the bacteria and you would plate them on a McConkey plate, and then what would you look for? These are gram-negative, so they'll grow on a McConkey plate, right? And then what you look for pink colonies, which indicates that they ferment lactose, right? So that is one of the most common uses for McConkey plates, where you have the city of Vancouver, for instance, has a microbiology lab, and they send workmen all over the city taking samples out of the water pipes to see, and then they bring these samples back to a laboratory, where microbiology technicians will streak the samples onto McConkey plates and then count the number of colonies that you see and how many of the colonies are pink, and then you will get something called a coliform index, and from that you can judge whether the water is contaminated or not. Okay, so these are the non-coliforms. They either have no ability to ferment lactose or incomplete lactose fermentation ability. So the enterics that we saw, the coliforms, the Enterobacter, Escherichia, and Klebsiella, if you put them on a McConkey plate, the whole area will turn pink. Whereas if you plate the other ones, you might get the colony being pink itself, but you won't see a pink area around it. Okay, so here, because you find these enterics inside the intestines of animals, it will be in their feces. The word feces and the word fecal mean the same thing. So fecal is an adjective, I guess. And so fecal coliform count is the number of coliforms that you find in a sample of drinking water, for instance, and you know that they got there as a result of that water being contaminated by feces. All right, now let me take a brief diversion to discuss water purity standards and how we do a coliform index or a coliform count. The protocol is you take 100 milliliters of a sample of water or a sample of milk, and then you do something called a serial dilution, which I'll explain in a minute. And then you put the sample onto McConkey plates, usually. Right, McConkey plates, and you count the number of colonies and you count the number of pink colonies. When you count the number of pink colonies, that will tell you how many coliforms there are in that water sample or that milk sample. Okay, these are the acceptable standards. These are the acceptable standards. Drinking water, you cannot, if you take a 100 ml sample and you test how many coliforms are in there, 0 is the cutoff line. If you find even one cell in 100 milliliters of water, that water is unfit to drink. It's unfit for human consumption. Okay, but there are other uses that we use, the other things that we use water for. We swim in it and we catch fish out of it. So for example, 200 coliforms in 100 milliliters of water is considered okay to swim in. You're just not supposed to drink it. Just try not to swallow any of it. Right? So Vancouver in the summertime, we have beaches. Our beaches, the water, I would not swim in the water in Vancouver, frankly, because it's very dirty. And anyway, there's some parts in the world where they have beautifully clean water, and the reason why and usually you find those places in the tropics. You find that beautifully clean water in the tropics because the sunlight has killed the bacteria in the water. Vancouver doesn't get much sunlight, and so therefore Vancouver water is very dirty. So I would not swim in the Vancouver water anyway. But if you must, you'll see that on the news reports they're constantly reporting whether it's okay to swim in the water or not. And in the summertime in Vancouver, every other day, the news on the evening news, they'll say you shouldn't be swimming in the water for a few days, and they'll put up signs on the beach, and so you shouldn't be swimming. And that's how they determine whether the water is fit to swim in or not. They do a coliform index on a sample of the ocean water, and it's very often dirty. Okay, what if you just want to go out and catch fish? You can have up to 1,000 cells in 100 milliliters of water. Right, so that's dirty, but you're going to wash the fish anyway before you cook it. You're going to wash the fish before you eat it, and presumably you're going to cook it well before you eat it, because otherwise you could catch. What would you catch? Which of the helminths is a common contaminant of undercooked fish? Right? Diphyllobothrium latum. Right? So you could get that if you don't cook the fish well enough. But it's okay to fish in water that has a coliform index of 1,000 cells per 100 mils. Okay. Now, a little interesting aside, some people are lactose intolerant. Now, in order to digest milk and cheese and other dairy products, milk, cheese, yogurt, you have to have enzymes that can break down lactose. So lactose is a disaccharide that is made of, what is it made of? Glucose and fructose. Oh no, I'm sorry, glucose and galactose. Sometimes I have to think of it myself. So lactose is made of glucose and galactose. If you have beta-galactosidase. That's an enzyme that will chop. Wait a minute now. I believe. Well, I can't remember the name of the enzyme, but you have enzymes that break lactose into glucose and galactose, and then you can directly metabolize the glucose in your mitochondria, and then there's another enzyme that inverts and rearranges the galactose so that it's glucose, and you can digest that as well. Okay, so when most humans eat milk or cheese, they have these enzymes and they have the ability to convert lactose into glucose and then digest it. Some people have defective enzymes, and so they can't digest the milk, and so it goes straight through it goes into their intestine, and it's not broken down and absorbed by the human intestine, which leaves it available for the symbiotic bacteria in our intestines to eat. And many of those symbiotic bacteria produce gases, particularly methane gas, and that's what makes a person flatulent. If they're lactose intolerant, they become very gassy. Flatulence, that's what that word down there means. Means gassy, and that is caused by the fact that they don't have the ability to break down lactose, and so the lactose is eaten by bacteria, some of which generate a lot of gas. So that's the origin of why people who are lactose intolerant sometimes get gas when they eat dairy products. Okay, so E. coli, we know all about that, but we need to learn about let's take a review of some of the virulent strains again. Okay, so a word about virulent or pathogenic strains of E. coli. Okay, every now and then you see on a news report, like this is shown on the bottom left here, there's a report on CNN saying that 2,000 people have become sick in 12 different countries across Europe due to eating carrots that are contaminated with E. coli. You see the word E. coli right there, right? See the word E. coli? Right, and so if you read that, you would think that that E. coli is bad, when in fact you and I know that E. coli is not bad. It's only the virulent strains of E. coli that are bad, and those E. coli, somehow you got a virulent strain of E. coli into the meat that makes a bunch of people sick when they eat hamburgers. Every now and then you'll see a news bulletin about a hamburger being recalled because it's contaminated with E. coli, or vegetables being contaminated with E. coli. Of course, we're not talking about regular E. coli. We're talking about the virulent strains of E. coli that are dangerous in small numbers. Any bacteria is dangerous in large numbers if you eat it, but generally if you eat small amounts of bacteria that are not virulent strains of that bacteria, it's fine. It's okay. Okay, so let's look at some of the virulent strains of E. coli. So we have enterotoxic E. coli, which is abbreviated ETEC, enteropathic E. coli, which is abbreviated EPEC, enteroaggrative E. coli, EAEC, enteroinvasive E. coli, EIEC, and enterohemorrhagic E. coli, EHEC. All right, so I may ask you multiple choice questions about these. Multiple choice questions. Remember, when you're studying for multiple choice questions, you don't have to memorize the thing completely. You just have to memorize it well enough to recognize it on a list of options, right? So that's why multiple choice questions are considered to be easier than short answer questions or essays, for instance, because you only have to memorize the names of these things well enough to recognize the name on a list. Right? Now, yeah, so that's a different kind of memorization, and because we consider it easier to recognize names on a list than to memorize them completely, therefore we feel that it's okay to ask you about a lot of different types of information, as long as we use multiple choice questions. And that's the attitude that I've taken in this course, which is that I can ask you about a lot of different things, and that's not an undue request, because all I'm asking you to do is to memorize all these names and all these things well enough to recognize them from a list. Right, so bear that in mind when you're studying these things, and try not to get too angry about the injustice of having to memorize all these things. I know it is actually difficult to do a lot of memorization, but not if you're just bear in mind that you're going to be recognizing things off of lists. Okay, so let's go through go through these virulent strains of E. coli. The only difference whenever you're memorizing these names, remember that the first and the last part of every one of these names is always the same. The first part is entero, and the last part is E. coli. Right? So the only thing you really have to memorize well enough to recognize is the middle part. Right? So enterotoxic is where the T in ETEC comes from. Enteropathic is where the P comes from. Enteroaggregative, aggregative is where the A comes from. Invasive is where the I comes from. And hemorrhagic, you know what hemorrhaging is. That means bleeding. Internal bleeding is where the H comes from in the worst of them, which is EHEC. All right, so let's look at these and why they are so virulent. Okay, so enterotoxic E. coli attaches. It has virulence factors that allow it to attach to the microvilli in the intestine, but it does not enter those cells, right? So it's not terribly bad because it doesn't get into the cells. But what it does is it secretes a toxin which causes those cells to produce a lot of, those cells to become leaky and to leak a lot of saline into the intestine, which causes watery diarrhea. So enterotoxic E. coli, ETEC, is not the worst, but it will cause diarrhea. Enteropathic, again, it has virulence factors that allow it to adhere to the microvilli in the intestine, and it actually effaces or scrapes the microvilli off. And if you scrape, so this is actually doing damage to the cells in the intestine. It's doing quite a lot of damage to the cells because it's kind of acting like a piece of sandpaper that's sanding or effacing the microvilli. And remember that the microvilli are there to allow you to absorb more liquid. And so if you can't absorb as much liquid, then you have, again, watery diarrhea. Okay, so this is an electron microscope image that shows the effacing that is caused by enteropathic E. coli. Notice up here we have this beautiful, looks like a shag carpet. These are the microvilli that you find on the inside of the human intestine. And down here, the bacteria has kind of scraped some of them away by killing off these microvilli so that the ability of the intestine to absorb liquid has been diminished, and so you get watery diarrhea. Aggregative, I have a great deal of trouble pronouncing that word. Right? It forms large aggregates of E. coli on the inside of the intestine, which blocks absorption, which again results in watery diarrhea. Enteroinvasive, it actually goes inside the cells. It has adhesion factors that allow it to attach to the microvilli, and then it goes inside through receptor-mediated endocytosis, which is a natural process, RME. You learned about that in Biology 120 or Biology 130. And once it gets inside, it damages the cells and it causes dysentery. So dysentery is diarrhea mixed with blood. So if you start damaging the cells, including the capillaries and blood vessels that surround the intestines, you will end up getting bloody diarrhea or dysentery. Okay. This is the worst of them all. It has the lowest. It has the lowest ED50 and the lowest. The lowest LD50 and the lowest ID50, and it causes hemorrhaging, right? And so particularly you'll have heard of a strain of E. coli called O157H7, right? So you do need to memorize this well enough to memorize it. O157H7 is a strain of E. coli that is enterohemorrhagic E. coli. It gets in, it attaches to the cells, but it doesn't enter, but it kills them by producing a very toxic compound called a Shiga toxin. And Shiga toxins are whenever you have a bacteria that produces a Shiga toxin, you need to memorize that as well. Shiga toxins generally cause diarrhea or dysentery. And so this causes hemorrhaging of the colon, so you have bloody diarrhea, very violent dysentery from enterohemorrhagic E. coli. You remember during the discussion on pathology that we did, the numbers for the lethal dose LD50 and the infectious dose, the ID50, for E. coli, EHEC, enterohemorrhagic E. coli like O157H7 around 10. So you only have to eat around 10 cells in order to get violent dysentery from that particular strain of E. coli. All right, moving on. Enterobacter, obesity, Klebsiella sometimes causes pneumonia. Not that common a cause of pneumonia because it's difficult for it to get, somehow has to get from the intestine into the lungs, possibly through the oral fecal route. You drink some contaminated water and you accidentally inhaled some of it. You could get pneumonia from Klebsiella pneumonia. Salmonella typhi, Salmonella genus, it causes typhoid fever. Here's a little historical anecdote for you. I will not ask you about this, but around the turn of the century, around the 20th century, around 1900, in the history of microbiology, this is a fascinating chapter in the history of microbiology because there were a number of typhoid outbreaks that occurred in New York. And you've heard with the coronavirus, the COVID-19 and so on, you've heard a lot of talk about tracing, infectious tracing. What that means is when somebody gets sick, you say, the investigators get to work and they say, where could you have caught that disease? And so who are the people that you've come into contact with? And, do you all have the same, do you all have a friend in common or something like that? And when they did the detective work tracing among these, in fact, the typhoid was, the typhoid outbreaks were spreading amongst wealthy families that had servants. And they found out that one of the things they all had in common was that at one time they all had a servant who was an immigrant from Ireland named Mary Mallon. And she acquired the somewhat insulting title of Typhoid Mary. So she was considered what we would call these days a patient zero. So the theory was that she brought typhoid with her from Ireland. And when she was preparing people's food, it got into their food. Now so she was called Typhoid Mary and she was famous. And one of the reasons why her case was famous was because it was the first time in the United States that they had to consider the idea of mandatory quarantine. So you know that the United States started out as being a very libertarian type of a country where they weren't allowed to imprison you unless you committed some serious physical crimes. And so Mary Mallon was spreading this disease, but she hadn't committed any crimes. But they said, well, you're spreading a disease. We can't let you run around loose. And so they forced her to be in the hospital until she was cured of the typhoid, until she was cured of the Salmonella typhi, right? Or until she got better from the typhi. So that was an interesting case in the history of the United States because it was the first time that they were ever talking about depriving somebody of their liberty based on a disease that they were carrying, as opposed to having committed a crime, for instance. All right, so here there is as I mentioned here in this slide, there is a vaccination that prevents you from getting Salmonella typhi, which you can take, but it's not common to get that vaccination unless you live in an area where Salmonella typhi is endemic. And as you can see from the map, North America Salmonella typhi is not endemic to North America anymore. It is endemic to these other parts of the world that are shown on the map. And so depending on where you live, it might be a good idea to get vaccinated against Salmonella typhi. It depends on where you live and whether there's a possibility of Salmonella typhi is spread through the oral fecal route, so it can contaminate water or food. If you live in any of these areas, you have to be concerned about it. But it doesn't necessarily mean you necessarily have to be vaccinated, although you can be vaccinated against it. Legionella's disease. As I said, you had a bunch of these old retired soldiers in 1976, it was, pardon me. 1976, they all came down with an unknown disease, or unknown respiratory disease. Many of them died. And after much investigation, they discovered that it was caused by a bacteria. It was a Bacillus, I believe, and then they named it in their honor, they named it Legionella pneumophila. And it turns out that Legionnaire's disease, Legionella pneumophilia, is easily spread through air conditioners that are cooled by water. So the hotel that all of these Legionnaires stayed in was contaminated with water that had contaminated with Legionella. Now that it seems, like that's something they would check, but every now and then, everywhere, all in all places all over the world, every now and then, central air conditioning units get contaminated by Legionella pneumophilia, including Vancouver. So the Guildford Walmart, which is in a suburb of Vancouver in Guildford, you can see by the date here, had an outbreak, had a little foci of Legionnaire's disease in the mall. So this was just a couple of years ago. And so it was caused by dirty air conditioners, a dirty central air conditioning system in the Guildford Walmart. All right, so they had to clean it out, close it, clean it out, and then reopen it. Okay, Pseudomonas aeruginosa causes wound infections. Right, so it's an opportunistic infection. Pseudomonas aeruginosa has some necrotic enzymes, which means it kills the tissue, and then the bacteria kills the cells and then spreads. Right, and then it causes a kind of a disgusting infection of the wounds. If you're into such things, and you're curious to see what it looks like, I'm not going to show you because it's quite disgusting, but you can Google Pseudomonas aeruginosa infection of wounds, and you'll see the most disgusting looking infected wounds you've ever seen. And as I mentioned, one of the problems is that it actually that one of the cleaning agents that hospitals sometimes use are a group of compounds called bisphenols, and another group of cleaning agents, another group of chemicals called biguanides. So bisphenols and biguanides kill many different types of bacteria, with the exception of Pseudomonas aeruginosa, which actually likes it. It actually encourages growth of Pseudomonas aeruginosa. Why do they still clean hospitals with these compounds? I don't know. They're supposed to know. The hospitals are supposed to have infection control experts, and they're supposed to know this, but apparently some of them don't. So you can, if you're ever in the hospital, you should ask them about that, making sure they're not using bisphenols or biguanides to clean the hospital. But if you go to the drugstore, you go to the pharmacy or the supermarket, you see amongst the cleaning products, you'll see these cleaning products that list the word antimicrobial or antibacterial soap on the label. Usually those soaps or those cleaning products contain bisphenols or biguanides, and so those are actually dangerous because they can spread infections. So if you have wounds, I wouldn't want to wash my hands with bisphenols or biguanides if I had a cut on my hand or something like that, for instance. So you'll also find biguanides and bisphenols in toothpastes and mouthwashes, and they advertise them as being antimicrobial mouthwashes, but that's true because they kill most bacteria, but not Pseudomonas aeruginosa. And we already know about Shigella. Shigella dysentery causes dysentery, and Vibrio cholera causes cholera, both of which are accompanied by diarrhea. Death is usually, if it's fatal, it's caused by dehydration. That's something that it only happens under certain circumstances. If you get cholera or you get Shigella dysentery and you're close to a nice modern hospital, they can make sure that you drink lots of good fresh water that doesn't have these contaminants in it, and you can get over it. But if you're someplace else where there isn't, if you're in a very poor place, in a very rural place where they don't have access to fresh water, and you have no way to filter it to filter out the microorganisms, there could be a problem. So, but that's generally not the case. All right, Yersinia. The Yersinia genus is again a member of the gamma proteobacteria class, and it is responsible for example, in particular, Yersinia pestis killed about half the population of Europe during the Middle Ages due to something called the bubonic plague. If you're interested in European history and European literature and things like that, there was a lot of art and a lot of literature and stories and whatnot written in the Middle Ages and after the Middle Ages, which kind of revolves around, or the stories take place where there's a backdrop of bubonic plague going on. Uh, um, you might know, some of you that are art majors, you might know that there's an artist named Bruegel who famously painted a picture called The Triumph of Death, where that was meant to, that picture, that painting was about the bubonic plague. It's a famous painting. It's quite hideous. And as funny as this may be, when I was in the microbiology department at UBC. The head of the microbiology department was a very eccentric man named Julian Davies, who was a famous microbiologist. And he had a great big huge print, an oil reproduction of Bruegel's The Triumph of Death in his office, because it was a microbiology painting, presumably, but everyone thought it was kind of weird. So anyway, the bubonic plague was a big part of European history, a big major event in European history. So here we have some lithographs and paintings from the early Renaissance. And so on the left here we have a couple of people who have lumps all over their bodies. The lumps are referred to as buboes. What those lumps are they are swollen lymph nodes. Lymph nodes are part of the lymphatic system. So if the Yersinia pestis gets into the lymph nodes, they swell up and form these buboes, and hence the name bubonic plague. Right, and in the background here, there's an alchemist or a physician who's throwing herbs and spices into the air, mainly because they had no idea how to deal with this, and that was they speculated that maybe some herbs and spices might do something to cure this, but of course it didn't. And over here on the right, we have a lithograph, which is a common way of reproducing artworks in the Middle Ages, and you can see the evil angel of death. You can see the bubonic plague in the form of the evil angel of death dragging people away from their loved ones, dragging them out the door from their weeping families and so on. And this is all to do with the bubonic plague. So the bubonic plague was also known as the Black Death, and it was characterized by buboes, which are swollen lymph nodes, as I said. It was caused by the bite of fleas. The fleas were actually hitching a ride on rats. So it was believed for a long time that the bubonic plague was carried by rats. In fact, it wasn't carried by the rats, it was carried by fleas that were on the rats, and therefore Yersinia pestis is an example of a disease that has an arachnid vector. It has a flea vector, and not an insect, but an arachnid vector. So so that's yet another bacterial disease that has a vector. Okay, let's move on to the Delta proteobacteria. The only one that we're really going to learn about is a bacterial genus called Desulfovibrio. It's kind of interesting to the biotechnology industry. It is of interest to people who are interested in cleaning up toxic wastes that involve sulfur or sulfuric acid. So for example, there's been a lot of industrial activity in the world where we burn coal to make steel or do other things. And the waste from the emissions from the coal, from smelters, smelting furnaces and things like that, the emissions from the coal get carried up into the atmosphere, and they contain sulfur. And then that sulfur mixes with water when it rains, and the sulfuric and converts to sulfuric acid, and the sulfuric acid ends up in lakes. And so the pH of the lake drops, and then nothing can live in the lake, and so everything dies. So there's an interest in finding some easy way to get rid of sulfuric acid in lakes as a result of what's called acid rain. And so one of the possible things that they're thinking about doing is just taking these lakes and throwing in a bunch of desulfovibrio in order to convert the reduce the sulfuric acid back into sulfur, and then clean up the sulfur powder or something like that. So there is some interest in desulfovibrio, which is a member of the Delta proteobacteria. Now let's look at the Epsilon proteobacteria. We're only going to learn two members, two genuses, two geni from the Epsilon proteobacteria. One is Helicobacter pylori, which you've already learned about. Helicobacter pylori causes ulcers in the stomach. In rare cases, Helicobacter pylori can even cause stomach cancer, but that's rare, happens less than 1% of the time. And the other one is another microorganism that causes gastroenteritis or gastritis, and that's Campylobacter jejuni. Campylobacter jejuni is also an epsilon proteobacteria. Now, both Campylobacter jejuni and Helicobacter pylori are spirochetes, and they are flagellated spirochetes. So they have flagella, but they're arranged slightly differently. So they are enterics, and they are also spirilla. On the left, we have an electron microscope image of Campylobacter jejuni. Take notice of the fact that it is a monotricious spirillum, a monotricious spirillum. So I might ask you that in a quiz on an MCQ question. And on the right, we have a false color SEM. So that means, SEM, scanning electron microscope images, or scanning electron microscope micrographs, as they're called, are in black and white, but occasionally somebody will color them in artificially just to make it easier for people to see what's going on. Right, so notice that the Helicobacter pylori, of course, is a lophotricious spirillum. Right, so we have Campylobacter jejuni is the only example that I'll teach you of a monotricious spirillum. The other monotricious bacteria that I told you about was Vibrio cholera, which is a Vibrio, not a spirillum. And then Helicobacter pylori is a lophotricious spirillum. Okay, I'm going to show you a slightly disturbing image in a second. First, we'll look at a cartoon image of the stomach. Okay, so this is just demonstrating that there's Helicobacter pylori up here making its way into the stomach, often through food. Somebody eats some food that has a little bit of Helicobacter pylori in it, and Helicobacter pylori likes to eat mucus, and there's a nice thick layer of mucus that protects the stomach wall from hydrochloric acid that's made in the stomach. By the stomach, and it eats away at the protective mucus layer, and then you get these ulcers burned into the stomach. Okay, now I'm going to show you a slightly disturbing picture of the human colon when it has infections of Helicobacter pylori. Okay, so on the far left we have a normal colon, and that is what you see if you have an asymptomatic or a subclinical infection of Helicobacter pylori. Then next to that we have more than 80% of the infections for Helicobacter pylori have no symptoms. They're subclinical infections. Then if you move to the next picture, in 15 to 20% you have some mild gastritis, right? So there's a little bit of irritation of the colon. And then in a few cases you have a duodenal or a gastric ulcer. Gastric ulcer is a blister or an ulcer in the stomach, and then a duodenal ulcer. Those of you that took Biology 130 know that the duodenum is the first part of the small intestine that makes contact with the stomach. Okay, and then we have, in less than 1% of the cases on the far right, we have a polyp, which is a stomach cancer, basically, or intestinal cancer. All right, let's look at other gram-negative phyla, important gram-negative phyla. Okay, the spirochetes are very thin, wiry, kind of spring-like organisms. They have, instead of having flagella on the outside, they have a bundle of flagella on the inside, something that if you have a bundle of flagella on the inside of the cell, it's referred to as an axial filament. So you need to know which types of bacteria have axial filaments and what an endoflagella is. Endoflagella, of course, is just flagella that are on the inside of the cell instead of the outside. And based on what you know about things like the glycocalyx, for instance, you would know that flagella are made of protein, and so they are highly immunogenic. So the immune system can relatively easily kill bacteria that have flagella on the outside. But if the flagella is on the inside, the human immune system has trouble seeing it if it's on the inside like that. So it's another way to evade the immune system. Okay, another phyla that we're going to look at briefly is called the planctomycetes. They are harmless water organisms that live in the ocean or in freshwater lakes. They are not photosynthetic, but they are green anyway, and they are either they either sit in the same place and are sessile, what's called sessile, or they take off and they swim someplace. Right now, when they are sessile, the swimmers that are referred to as swarmer cells, meaning that they can swim around. But the sessile ones remain in place because they stick to a rock or some other surface at the bottom of the body of water. And they stick with the help of an organelle called a holdfast organelle, which we're going to look at. So the holdfast organelle is basically a suction cup on the end of a filament, which allows the planctomycete to remain stuck to a rock or something like that. Now, as such, because they can physically stick to rocks and other surfaces underwater, the planctomycetes are important to something called biofouling. So any of you that have ever had a boat, or you know somebody that has a boat, a little boat or something like that, you know that if you leave it in the water long enough, things will eventually grow on the bottom. And that process is called biofouling. And that process is begun often by planctomycetes, because planctomycetes have a special organelle, the holdfast organelle, that allows them to stick to these surfaces. Okay, and then finally, we do have some bacteria that are called the phototrophic bacteria, and they are actually photosynthetic. So they are capable of carrying out photosynthesis, harvesting energy from the sun and using it to build glucose. And we believe that these phototrophic bacteria were the original ancestors of chloroplasts in plants. So they were the origin of chloroplasts in plants. Sometime about 1 billion years ago, a eukaryotic protist swallowed one of these phototrophic bacteria, and instead of digesting it, it just stayed inside the cell, didn't make contact with a lysosome and get broken down. And as such, they kind of multiplied inside the cell and just stayed there, and the cell decided that they liked them anyway because they're producing lots of glucose. Okay, so looking at the spirochetes, as I said, they have an endoflagella. They have endoflagella. This is a plural, right? So the endoflagella, this is where they have a whole bunch of flagella that are kind of bound together. And that bundle of endoflagella is referred to as an axial filament, which is inside the cell. And so spirochetes are long and thin and flexible and shaped like a spring or like a corkscrew that you use to take the cork out of a bottle. They look sort of like that. And the two that we're going to only learn 2 of them in this course, and you already know what they are. Treponema pallidum causes, what does it cause? Do you remember? It causes a sexually transmitted disease called syphilis, which is also a teratogen, by the way. And Borrelia burgdorferi is a spirochete that causes a disease called Lyme disease, which is often mistaken for Bell's palsy. Bell's palsy is a neurological disorder that suddenly causes half of your face or sometimes half of your body to become limp, as if you were having a stroke. It kind of looks like you're having a stroke when you're not. Bell's palsy is mimicked by Lyme disease. Lyme disease is the disease that's carried by Lyme disease is caused by the bacterium Borrelia burgdorferi, and it is a spirochete. Okay, so on the left here we have Borrelia burgdorferi viewed under a bright field microscope, and on the right we have Treponema pallidum viewed with what? That is a scanning electron microscope image that has not been colored in, right? So there's a SEM photo of Treponema pallidum causing syphilis on the right. This is a cross section. So this is a transmission electron microscope taken where somebody sliced up the bacterium on a microtome and then mounted it on slides and stained with osmium tetroxide. So you can see this is the cytoplasm of the cell. And this little thing here is a bundle of flagella that are actually underneath the cell plasma membrane, and they're bundled together to form an axial filament. right? And that's what the bacterium uses to swim. So they can swim quite fast using this internal kind of a filament structure. Planctomycetes. Okay, so they have a flagella. They live in both saltwater, the ocean, and freshwater lakes. They are perfectly harmless. Most of the time, they are sessile, meaning that they stay in the same place, and they stay in place because they're stuck to the surface using something called a holdfast organelle. Now, as I said, they are involved in the process of biofouling. When you leave things in the water, they get coated with a layer of microbes, and the first ones to stick to that surface are usually the planctomycetes. A good example, memorize this name for multiple choice questions, is Colobacter crescentus. It's called crescentus because it's shaped like a crescent, which is actually a vibrio shape, right? So crescentus, it's crescent shaped. And so it's involved in biofouling. It's also it's of interest to people who are doing biotechnology applications. Okay, so this is what it looks like. There's the holdfast organelle. And the holdfast organelle is basically, as I said, it's a suction cup on the end of a stalk. So there's the stalk. And this holdfast organelle allows it to hold fast, to hold in place and stick to things. All right, this is an old friend of mine named John Smith. He's a professor at UBC, and he's one of the world's best experts on Colobacter crescentus. He's probably going to be retired by the time any of you graduate with your degree. But if you were interested and he's still working, you could ask him if you could join his lab as a graduate student or something. If you're interested in this, I could recommend you. And anyway, he has been working for many years on biotechnology applications for Colobacter crescentus and other planctomycetes. And so one of them is the fact that the holdfast organelle sticks to rocks and surfaces, but it has a very high affinity for metals, which nobody expected. Nobody knows exactly why, but it has a very high affinity for metals. So one of the applications that John is working on, for instance, is he's finding ways. We do gold mining here in British Columbia, and one of the tricks to gold mining is you have to find some way to settle. You have to find some way when you dig the gold out of the ground. You have to find some way to separate the gold from the dirt, and there's a complicated process to do that. But John has this idea that he's been experimenting with, where you mix the gold and the dirt with water and Colobacter crescentus. The holdfast organelles will latch on to all of these little gold particles and make them float. And then the dirt will just sink to the bottom. And then you can easily separate the gold from the dirt. Right? So that's one possible application. Uh, I know that John also had a grant where he was looking at Colobacter crescentus fouling submarines and getting stuck to submarines. And as I said, so biofouling is something that happens, and the process of biofouling aquatic vehicles like boats is usually begun by planctomycetes. Okay, phototropic bacteria. Two of them that we're going to learn about, two phyla that we care about. One is called the cyanobacteria. The cyanobacteria is a phylum that appears kind of blue-green in color. They are oxygenic, meaning that they have the ability to create oxygen using photosynthesis, right, the way plants do. And they're kind of blue-green in color, right? And we believe that some of those were the early precursors, the early ancestors of chloroplasts in plants. And then there's the phylum Chlorobi, which is green sulfur bacteria, which has the ability to reduce sulfur or reduce sulfuric compounds. And it is actually of potential use in biotechnology to clean up sulfur spills in lakes and things like that. Okay, so you can see the purple bacteria. This is a test tube containing some of each. So the green sulfur bacteria is this green stuff. The purple bacteria is this purple stuff. Now it grows in lakes and freshwater lakes and so on. So occasionally you will find, for whatever reason, one of these bacteria will get overgrown in a freshwater lake, and you can see here this lake is totally green, and the water is no longer transparent, it's opaque, and that's because there has been something called a bloom of these cyanobacteria. All right, let's look at the gram-positive bacteria. So as we said before, there are many ways that you can classify bacteria. The most common way is by asking whether they are gram-positive or gram-negative. That's one way. And then in addition to that, you can also classify them into different categories based on the amount of GC pairs that they have in their genome. So as you remember, most organisms have around 50% of the time, 50% of the genome is AT pairs and 50% is GC pairs. Remember that AT pairs are held together by 2 hydrogen bonds and GC pairs are held together by 3 hydrogen bonds. Now, some bacteria and there are other organisms on Earth as well besides bacteria. There are some organisms on Earth that have more than 50% GC pairs, more than, and some that have less. And so that's another way that we can classify all the living things on Earth by the number of GC versus AT pairs that make up the genome. And as it turns out, that's a relatively easy test to do. It's very easy. It's very easy to determine what percentage of the genome is made up of GC pairs versus AT pairs. In fact, what you do is you put the DNA into a test tube and then you heat it up, and you know that the DNA is a double helix, so it's 2 strands that are held together by hydrogen bonds on the inside. You put that into a spectrophotometer and you shine a light through it, and then as you're heating it up, the properties of the light going through the test tube will change as the double helix, which looks kind of like a zipper, as that zipper unzips, because as you heat it up, you will break the hydrogen bonds and the 2 anti-parallel strands of DNA will come apart, just as if you were unzipping a zipper. And so the temperature at which that happens, you can measure that. And as it happens, that because GC pairs have 3 hydrogen bonds and AT pairs only have 2, if you have an organism that has a high GC content in its genome, the DNA will melt or unzip at a higher temperature. And so that's an easy way to tell the GC content of an organism. And also the other thing is that organisms that live in hot environments, thermophiles, tend to have a higher GC content in their genome for specifically that reason. Because if they live in a hot environment, they don't want the heat to cause their DNA to melt. And when I say melt, by the way, that's a technical term. That's a term of art that means it unzips, it comes in half. And so it's also, for those of you that have taken Biology 200, that's also what I mean when I use the word denature the DNA. Right? So if you're a thermophile and you live in a hot environment, you don't want your DNA to be coming apart, to be denaturing as a result of the heat. One way to prevent that is just to have more GC pairs in your genome. All right, so we can also divide bacteria based on how many GC pairs they have. All right, so we're only going to study one phylum that has a high GC content and is also gram-negative. Gram-positive. It's written right there, right? So now we're into the gram-positive bacteria that have a high GC content. The only phylum we care about are the Actinobacteria. Okay, so here are some of the important members of the phylum Actinobacteria. All Actinobacteria are gram-positive with a high GC content in their genome. Okay, starting with the genus Frankia, example, Frankia californiensis, right, it's another nitrogen fixation bacteria that allows plants to fix nitrogen. So the bacteria lives in the root nodules of plants and then sucks nitrogen out of the air and converts it into organic nitrogen, ammonia, and amino acids. And so the genus Frankia is an actinobacteria that is very good for us. It helps us to grow our crops, to cultivate legumes, basically. Right? Okay, now we go on to the bad ones. The genus Corynebacterium, example Corynebacterium diphtheria, causes diphtheria, which is spread by coughing. and it's characterized by a fever, a cough, and a swollen throat. There is a vaccine against diphtheria. Generally, you don't take the vaccine unless you live in an area where diphtheria or diphtheria is common. Okay, next we have the genus Mycobacterium, which is very important. As you remember, mycobacteria are also known as acid-fast bacteria. They're generally bacilli that are acid-fast, meaning that they have mycolic acid in their cell wall, and you have to use a special staining technique to see them, basically. So you use a stain. Do you remember which one it is? Right. The stain is carbylfusion. Right, so you stain these bacteria are gram-positive, but it's very difficult to tell because the crystal violet has a hard time getting through the mycolic acid in order to stain them. So if you stain some cells and you're looking to see if they are gram-negative or gram-positive and the staining process doesn't work very well, then you'll suspect that they might be acid-fast bacteria, that they might be mycolic acid bacteria. And so then you use carbylfusion, and then you tell that, okay, this is one of those. It's one of the Mycobacterium genus. Okay, so there are two that we're going to learn about that you already know about, basically. One of them is Mycobacterium tuberculosis, which causes a disease called tuberculosis. Tuberculosis is spread by coughing. It's spread by droplet transmission from coughing. It preferentially infects the lungs, and it can actually damage the lung cells, causing you to cough up blood. If you're sick or infirm or elderly, this could potentially be deadly. So, as I mentioned earlier in the course, Mycobacteria tuberculosis, if you get a tuberculosis infection, it's accompanied by a mandatory quarantine. That means you go to the doctor with a cough, and they say, unfortunately, they run a test on you, and then they say, unfortunately, you've got tuberculosis, you have to go to the hospital, and furthermore, you have to give us a list of everybody that you've been in close contact with, and then we have to test them to make sure that they don't have it. If they do, they have to go into the hospital with you, and then we have to get a list of everybody that they were in contact with. Right, so it happens every now and then. A couple of years ago, we had a student at Columbia College who had tuberculosis, and everyone that was in the college had a record of every class that they were in, so they contacted everybody who was in the same class with this guy, and they all had to be tested. Fortunately, nobody else did have tuberculosis. Right? And occasionally, this is just the nature of people being in the same place. Occasionally, we do have one or two people who get into Columbia College who come out with the measles. Right, and so they haven't. That means that, and then we have to do contact tracing to see who they've been in contact with, and inform everybody that was in a class with them that they should be checked for the measles. This is obviously the result of somebody not being vaccinated for the measles when they were a child. So we mentioned the measles, for instance, is a communicable virus. It's a virus, highly communicable, highly contagious virus that can be prevented by a vaccine that most people get when they're about 13 months old. Right, so most people get the measles, mumps, rubella virus. In one single vaccination, you're vaccinated against measles, mumps, rubella, and varicella zoster, and at the age of 13, so then you're not in danger of catching any of those diseases later on. But occasionally somebody's not vaccinated and they catch it, and then if they contaminate other people, you have to worry about whether those other people had vaccinations. And if most of them did, the virus doesn't spread very far because you're vulnerable to it, but not everybody is. In fact, most of the people are immune to that disease because they were vaccinated, and that's what we call herd immunity. The larger group of people, most people are immune to it. So even if one or two people get the disease, it won't spread very far because most people are immune to it. Most people were vaccinated to it. That's called herd immunity. All right, so Mycobacterium tuberculosis is a very contagious bacteria spread by coughing, and it causes a lung infection called tuberculosis. The other mycobacteria, which again is an actinobacteria with gram-positive with high GC content, is Mycobacterium leprae, which causes the disease called leprosy, which is generally spread by touching, not coughing. And it causes granulomas, which are lumps. And also ulcers. So ulcers, blisters on the surface of the skin, but also lumps underneath the surface of the skin called granulomas. Okay, finally a rather mundane one down here at the bottom. Propion, Propionibacterium, example, Propionibacterium acne, which causes acne on the face. The sebaceous glands and the hair follicles on the face sometimes get clogged, and these bacteria like to live in the follicle and eat the proteins that are produced, the sebaceous fluids that are produced. Okay, let's take a couple of minutes and discuss Mycobacterium and Mycobacterium tuberculosis and Mycobacterium leprae. Okay, so they are acid-fast bacteria that have mycolic acid in their cell wall. If you want to see them properly, you have to stain with carbonfusion. Okay, so there are two. They are intracellular parasites that provoke the formation of, it provokes a reaction by the immune system, forming these lumps that are called granulomas, right? Granulomas, and also occasionally lesions that produce sores, right? So there are Mycobacterium tuberculosis is particularly suited. It's spread through droplet transmission, and it's particularly suited at colonizing the lungs. So it's a respiratory disease. And then Mycobacterium leprae is spread by direct contact, skin to skin, through touching, and it causes leprosy. Okay, looking at tuberculosis first, Mycobacterium tuberculosis causes tuberculosis. It causes granulomas in the lungs, and when you have these lumpy structures in the lungs, they are named tubercles, right? So tubercles, hence the name tuberculosis. If it spreads, it could spread throughout the body and cause meningitis, which is sometimes fatal, and that's one of the reasons why it's accompanied by a mandatory quarantine. You can't have somebody going around spreading this disease, even if it means infringing on their liberty. Okay, so here's some lungs. These lumpy things inside are the tubercles, right? So you get these bacteria into the alveoli of the lungs, and they form colonies inside the lungs, occasionally causing lumps called tubercles to form. Here's an X-ray showing somebody with tubercles in the lungs. Okay. Now, Mycobacterium leprae causing leprosy causes two things to happen. This particular bacteria likes to live on the skin. It is what they call epitheliotropic. Epitheliotropic means that tropism refers to what body tissues does this microbe like to live in, and it happens to like living in epithelial cells, but it was epithelial cells of the skin in particular. So it infects the skin through touching. If it gets inside the body, which it does occasionally, it's as an intracellular parasite, it likes to live inside Schwann cells. Those of you that took Biology 130 know that Schwann cells are cells that produce myelin, which is wrapped around motor neurons and sensory neurons that travel up and down the spine. So if the bacteria gets in there, it will cause nerve damage. Right? It'll cause nerve damage, and so one of the symptoms of leprosy is that in addition to the scarring from the lesions and the granulomas, is that you start to lose sensation in your fingers and your toes and your hands, and so lepers sadly many times injure themselves by touching things touching things that are damaging to the skin or whatever. You touch a hot stove or something by accident, or you touch something that hurts, that would normally hurt somebody, but you don't have as much sensation in your hands or your feet, so you can do damage to yourself that way. Okay, now down here at the bottom, I wanted to draw your attention to the fact that there are two types of leprosy. One of them is called tuberculoid leprosy, and one is called leprotomous leprosy. So notice that leprosy causes granulomas, which are the lumps on the skin, as well as lesions of the skin. Lesions of the skin, of course, are blisters. Okay, now the blisters are the most infectious part, right? So if you touch somebody who has leprosy on the skin where there's no blister, you probably won't get leprosy. But if you touch the blister, if you touch the sores, the lesions, you probably will. So tuberculoid leprosy is just a variation of Mycobacterium leprae. The way it works out, your body ends up having more granulomas and fewer lesions. versus leprotomous leprosy, where your body has more lesions and fewer granulomas. As a result of that, because it's the lesions that are infectious, that means that tuberculoid leprosy is a little bit less infectious than leprotomous leprosy, but not much. There's not much of a difference. All right, so leprosy again is a big part of world history, ancient history. Like in the Middle East, we have all these stories about lepers going around, and leprosy was a disease that was endemic to the Middle East. And those of you that are interested in the history of Europe and the history of the West and so on, you know that in the Middle Ages, in the 12th and 13th centuries and so on, the Christian kingdoms of Europe went on what was called the Holy Crusades, which was a war basically to try and take Jerusalem back from, I believe it was from the Ottoman Turks, Muslims. And when they were doing that, in the process of doing that, they picked up leprosy and they brought it back to Europe. So they had leprosy in Europe from then on. So here's a typical leper from the Middle Ages, from the Dark Ages. This is an old lithograph from a book. And you see that the leper is covered in, has lesions on his face, right? And he's carrying a bell. It was the law that if you were a leper, you had to carry a bell around and you had to ring the bell when you were walking around so that people could get out of your way and they wouldn't be contaminated by you. And of course, what they used to do was they would sometimes send all the lepers to a leper colony where they would all live without danger of contaminating or infecting anyone else. So that again was, I suppose, that was the Middle Ages version of a mandatory quarantine, right? So leper colonies, that was a thing. They don't really exist anymore because we can cure. It's a bacteria, so we can cure leprosy with antibiotics. And so as soon as antibiotics became fashionable, then leprosy in many places, most places in the world, leprosy became a thing of the past. Okay, so they live inside Schwann cells. Here's a Schwann cell that's wrapped itself around the axon of a neuron. So if they get in there, they can cause neurological damage, particularly to sensory neurons and motor neurons. Okay, I mentioned the fact that tuberculoid leprosy is slightly less contagious than leprotomous leprosy, and so it is sometimes called dry leprosy because it has fewer lesions. The lesions are runny and leaky, so it's called dry leprosy, but and there's a myth, an urban myth that dry leprosy is not contagious. That's a myth and it's not true. Just as a little aside, this is a little personal anecdote. I'm not going to ask you about this, but this was a book that I read when I was a boy. It was popular when I was a boy. It was a book that was newly published by a Frenchman named Henri Charrier called Papillon. Henri Charrier was a gangster in Paris in the early 20th century, and he was a tough gangster that had a tattoo of a butterfly on his chest, and the French word for butterfly is papillon. So he was a criminal, and he was sent to a penal colony, a French penal colony in French Guiana called Devil's Island, and then he claims, and he wrote a book about his adventures living in this prison colony called Devil's Island in French Guiana and escaping to a leper colony, and he lived for a while among the lepers, he claims. And why did he not get leprosy if he lived among the lepers? He claimed he didn't get leprosy because the leader of the leper colony had dry leprosy, and that leader was the only one who touched Charrier's food. And so this leper with dry leprosy made this guy food, and he gave him things and gave him clothes to wear and stuff like that. And he was not contaminated by the leprosy because this guy had dry leprosy. By the way, that book was made into two different movies, both of which are quite good. But it was a famous story, right? And it was a famous story, caused a lot of sensationalism in the 60s and 70s. But there was always controversy about how truthful Charrier was being about his adventures. And so now you, as microbiologists, know that he probably was not telling the truth, or he was probably embellishing the truth. He was exaggerating, because you know that there's no such thing as a non-contagious leper. You know that dry leprosy simply means that you're less contagious than leprotomous leprosy. Tuberculoid leprosy is slightly less contagious. Okay, so that debunks Charrier's book. That has nothing to do with anything other than a historical anecdote, a colorful anecdote. I'd never ask you about that on a test. Okay, so Mycobacterium tuberculosis, Mycobacterium leprae. All right, let's look briefly at the Firmicutes. Okay, so the Firmicutes are gram-positive bacteria that have a low GC content. The most famous one that we've talked about so far in this course is the genus Clostridium, which has four members that we need to know about, that you need to know about: Clostridium botulinum, Clostridium difficile, Clostridium tetani, and Clostridium perfringens are four species of Clostridia that you need to memorize for this course because they are pathogenic bacteria that cause a lot of human damage. They are all soil anaerobes, so they all like to live in the soil, and they all are damaged to various degrees by oxygen, some more than others, right? So they are anaerobes that live in the soil. Okay, next, the genus Lactobacillus. We've already know about that one. It's a gram-positive bacteria that lives among other places in the vagina. It's a part of the vaginal microbiota. And it is also one of the bacteria that gets put into probiotics. So we heard about the fact that you can get hospital-acquired diarrhea if you take antibiotics that kill off all of the good bacteria in your intestine, thus allowing Clostridium difficile to enter. Right. Whereas if you get those antibiotics, every time you take antibiotics, you take some probiotics to put good bacteria back into your intestine. That includes Lactobacillus acidophilus. And then finally, there is actually a genus called Bacillus, and that includes Bacillus anthracis and Bacillus cereus. Bacillus anthracis, of course, causes anthrax because now members of the genus Bacillus produce endospores, and there's several of them. So they all produce endospores. So the endospores in Bacillus anthracis get tangled up in the hairs of sheep and horses, and they can cause anthrax in the humans that handle the wool or the hair from a horse. And Bacillus cereus is a common contaminant of grains and cereals, most commonly of rice. Right? So if you ever got an attack of food poisoning by eating stale rice, it was probably because it was contaminated by endospores of Bacillus cereus, and those endospores germinated, and then you had Bacillus cereus growing on the rice. Okay, so Clostridium. A few words about the Clostridium species, various Clostridium species. They are gram-positive bacteria with a low GC content. They all produce endospores. They are all obligate anaerobes, more or less. They're damaged by oxygen to a certain extent. They live in the soil, so therefore we have a soil reservoir for these, and they are usually pathogenic to humans. Okay, so Clostridium botulinum produces an exotoxin that paralyzes nerves and causes flaccid paralysis. Clostridium tetani, so that disease, of course, is called botulism. Clostridium botulinum causes botulism. Clostridium tetani causes rigid paralysis because it produces a neurotoxin that doesn't allow the muscles once the muscles flex, they are never allowed to relax. And so, in the case of, so the disease that's caused by that is called tetanus, and in both cases, it's usually spread by botulism is eaten. You eat food that's contaminated by Clostridium botulinum. The Clostridium botulinum got into the food through endospores. In the case of tetany, it causes a disease called tetanus. It usually got there because you cut yourself with a knife or some other sharp implement that was soiled with dirt. Clostridium perfringens gets into wounds again from dirt, and it causes gas gangrene, something called gas gangrene. This is where the tissue, you have necrosis of the tissue. The skin around the wound will turn black and die and turn black and get eaten by the Clostridium perfringens because that's what it's doing. It's sending out a lytic exotoxin. And then Clostridium difficile gets into your food in the hospital if you have somebody brings you a basket of carrots to eat while you're in the hospital or something like that, and they grew the carrots in their own garden, but they didn't do a very good job of cleaning them. And then you're on a course of antibiotics, which is killing all the E. coli and lactobacillus in your intestine, and then that gives the Clostridium difficile a chance to colonize your gut. Right? So the Clostridium difficile is normally excluded from the intestines through competitive exclusion, right? So they don't normally colonize the human gut. Okay, I'm going to show you a few images here that are slightly disturbing. All right, so Clostridium botulinum causes flaccid paralysis. It also causes sudden infant death syndrome, SIDS, sudden infant death syndrome. I'll explain that in a minute. What often happens is that it's in the soil, but the spore, it produces endospores which may germinate in canned fruit or jams, bottled jams that are under anaerobic conditions rather. Now, the bacteria itself is not that bad for you. The reason why you get botulism is because you eat the toxin, right? So in the case of Clostridium botulinum, you have a can that's contaminated, a can of food that's contaminated. It produces a lot. The spores germinate inside the can. The bacteria grow and release a lot of the Clostridium botulinum toxin into the food which you eat, and that's what causes the muscle paralysis. However, occasionally, now if you eat Clostridium botulinum endospores, they will probably not stay in the intestine long enough to germinate. So you don't have to worry about getting killed by eating the spores per se. It's the toxin that's deadly, right? However, if you're a baby and your gut has not yet been colonized by E. coli and Lactobacillus acidophilus and other things like that, it is possible for the Clostridium botulinum spores to germinate in your gut and therefore take over the intestine and thus produce enough of the toxin to kill a baby. So sometimes if a baby dies and they don't know the cause, they call it sudden infant death syndrome. There are several causes, but one of them could be Clostridium botulinum, which was caused by feeding a baby some food that had Clostridium botulinum spores in it. All right, so if you see a can like this, you never open it and eat the contents because this can is filled with Clostridium botulinum. The endospores got into the can, and then normally the fact that there's no air in the can would prevent other bacteria from growing. That's why you can it, to prevent aerobic bacteria from growing. But there are a few bad guys. There are a few bad bacteria that can grow in a can without air. In fact, they prefer not having the air, and so Clostridium botulinum is one of those. Okay, now some people accidentally kill their baby by dipping the baby's pacifier in homemade honey. Homemade honey often, or not often, but sometimes contains Clostridium botulinum spores, endospores. If you're an adult and you have an intestine that's filled with E. coli and lactobacillus, it doesn't matter because the Clostridium will not be able to catch hold and set up house in your intestine. But if you're a baby, it might, because babies have not yet had time to develop an intestinal microbiome. Okay, Clostridium tetani causes rigid paralysis, also known as spastic paralysis, and sometimes called lockjaw, because if the jaws, if the muscles in your jaw clench shut. Now, the technical term for this rigid paralysis is called opisthotonos. Opisthotonos. Right, and the route of entry for Clostridium tetani is the parenteral route. So the parenteral route, you get cut with a dirty knife or a dirty gardening instrument, like a dirty rake or something from the garden. The toxin that it produces is called tetanospasmin, right, and is abbreviated TENT. All right, so the history of lockjaw and tetanus is kind of a mixed history. In the barbaric old days, when people used to fight, men used to fight wars with swords and spears and sometimes bayonets. It's very common for soldiers to get minor wounds on the battlefield, and then 2 days later, they would get this. They would bend into an arch like that because every muscle in their body would flex, and then it would stay flexed and never relaxed, and you can't breathe. That condition is called opisthotonos, and then they would die. So in the old days, people who fought or got minor injuries in wars would end up dying for injuries that today can easily be cured because they didn't know about Clostridium tetani. All right, I'm going to show you an image of gas gangrene. This is where you have a wound that gets a few endospores that germinate under the skin, and that's enough to cause, because Clostridium perfringens produces a lot of exoenzymes that destroy the tissue. This is enough to give you gangrene, tissue necrosis. So this is gangrene of a wound, right? You have to, uh, by the time it gets this bad, there you have no choice but to chop that limb off because it'll just keep on spreading otherwise. And so that was the reason why in the medieval days and all the way up until the, like the Napoleonic Wars and things, if somebody was wounded in the arm or the leg, it was normal to cut it off because they didn't know how gangrene started. Right? So they didn't realize that you had to keep the wound clean, and you had to keep wounds clean because they didn't realize that gangrene was spread by a bacterium in the early 19th century. They did by the time they got to the late 19th century, but in the early 19th century and before, they didn't know that. So that the thought was that if you were wounded even slightly in one of the limbs, it was better to cut the limb off than to risk sewing it up and then having it become gangrene. All right, Clostridium difficile is the cause of antibiotic-associated diarrhea. All right, so you take a course of antibiotics either in or out of the hospital, and then suddenly you get diarrhea. What happens is that Clostridium difficile produces exoenzymes and exotoxins that damage the intestine, cause ulcers in the intestine, and those ulcers will kind of seal over with a kind of a pus that looks, it's kind of a scab basically. The ulcers in the intestine will cover over with a scab, and that is referred to as pseudomembranous colitis because the blisters inside the intestine are covered by something that's not quite a membrane. So pseudomembranous colitis, and then colitis means inflammation of the colon. Right, so I'll show you a picture of what that looks like. So brace yourself. Okay, so here's a warning. You often have outbreaks in hospitals of C. difficile outbreak due to this. And this is what a pseudomembranous colitis looks like. This is where you have a bunch of holes and blisters in the intestine that have been covered over by pseudomembranes. So there they are, the four bad guys of the Clostridia genus, this Clostridium genus: Clostridium botulinum, Clostridium tetani, Clostridium perfringens, and Clostridium difficile. All right, we already know about Lactobacillus acidophilus. And now, Bacillus anthracis and Bacillus cereus. Okay, so a few words about these two. Recall that Bacillus anthracis and Bacillus anthracis is a streptobacillus. It forms strips, right? It produces three exotoxins that are called protective antigen, or PA, edemal factor, which causes edema or fluids, swelling and fluids, and lethal factor, which causes cell death, which is called cell necrosis, right? So it is an aerobic organism which lives in the air, right? And it produces endospores, right? And and it was frequently, or once in a while, it would contaminate. You would get animals. So this is a zoonotic infection, where you would have people who tended to animals, in particular sheep, and the sheep would get infected with anthraxis on their skin, but there's not a lot to eat on the skin of a sheep, so the bacteria would start to starve, and under starvation conditions they produce endospores, and those spores would get stuck in the sheep's wool, and then the people who would shear the sheep and then spin the wool into yarn would get this disease on their hands, and it was called wool sorters disease, and that's how they got it. Also, people who would tend to horses and things could get anthrax as well. Okay, so they called it's a zoonotic infection. Anthrax is a zoonotic infection. It is sometimes caught by people who tend or take care of horses, cows, or sheep in particular. Hence, it was called wool sorters disease. You would sometimes catch it from shaving brushes. These are shaving brushes at the top, and then other horsehair brushes at the bottom. Or wool sorters disease, people turning sheep's wool into yarn. Okay, so the protective antigen, it produces three exotoxins that contribute to the production of sores, blisters, and sores, which is what anthrax does. So now the edema factor causes the blisters to become very runny, not with blood, but with serum and lymph fluid. So we call those weeping sores. Remember, to weep means to cry and make tears. So you have these sores that are kind of runny and produce a lot of saline, and that's the result of this protein called the edema factor. So edema means swelling and fluids. So it occasionally, Bacillus anthracis would occasionally cause a disease called hydrothorax, which is where if you manage to get anthrax inside the membranes that surround your lungs, those are called the pleural membranes. You could get something called hydrothorax. And hydropericardium is where you have fluid in the pericardium that surrounds the heart. And those of you who took Anatomy 130 know that can lead to a disease called cardiac tamponade. This is where the heart doesn't have room to fill up with blood because there's all this fluid in between the heart and the pericardium. Okay, so the lethal factor actually kills tissues and causes sores and blisters that are called eschars, right? So I might ask you which bacteria causes eschars, right? And that would be Bacillus anthracis, right? Sorry, I should have warned you, right? So it also produces phospholipase C and causes these eschars, which are weeping, runny sores. Now, there is a particularly nasty disease called inhalation anthrax, and it's one of the characteristics of wool sorters disease. So, cutaneous anthrax is if you have wool sorters disease and you have specifically cutaneous anthrax, the word cutaneous means skin, right? Cutaneous means the skin. If you have cutaneous anthrax, it means you have eschars or these runny blisters on your skin. If you have inhalation anthrax, that means you inhaled some endospores and it causes runny blisters on the inside of your lungs. If you have one or two runny blisters on the inside of your lungs, it causes pulmonary edema. That means your lungs will start to fill up with water, which will choke you. If you inhale a lot of endospores, then that will kill you, and it will be a very unpleasant way to die because your lungs just fill up with liquid from the inside, and there's nothing you can do about it. So it is usually deadly, and it's a very unpleasant way to die. But normally you would not, under normal circumstances, you would not inhale that many spores. However, some maniacs have seen fit to turn anthrax spores into a biological weapon of terror, basically, where you deliberately grow up a lot of anthrax and then you induce those anthrax cells to produce endospores by starving them, and then you dry them out and grind them up in a mill until you make a fine white powder that's made of nothing but anthrax spores. And then you mail that in a letter to somebody, and they open the letter, and this white powder falls out, and then they've inhaled thousands of spores, and then they could die of inhalation anthrax because there's so many spores in the same place, which you wouldn't normally find under normal circumstances. And then finally, gastrointestinal anthrax. This is where you swallow a few spores. Big deal. You might get one or two bleeding ulcers in the intestine, but that happens all the time for other reasons, and so that's not terribly deadly. So you remember during when we were doing the pathology section and we were looking at the LD50 values. You remember that inhalation anthrax had a fairly small LD50 value, so it's a particularly dangerous type. Okay, so as I said, inhalation and anthrax has been turned into a biological weapon by some maniacal people. Right, so here, you remember 9/11, September 11th, 2001. There was a terrorist attack on the World Trade Center in New York, in the United States, and right after that, so we know who did that, right? So we know that was done by Muslim extremists. Right after that, politicians in the United States started getting letters that contained anthrax. So here, for example, is a letter to Senator Daschle. Tom Daschle was the Senate, I believe he was the Senate Majority Leader in the year 2001. And so this letter contained anthrax. So you see somebody's got it in a laminar flow hood because they're basically disarming it. And so a few politicians in the United States got these letters that were filled with anthrax. And inside, you would see this letter that says in kind of broken English, you cannot stop us. We have this anthrax. You die now. Are you afraid? Death to America, death to Israel. Allah is great. All right, so they say this, and so we're supposed to believe that Muslim terrorists are doing this. In fact, it was done by a Caucasian Christian, right, nutcase, a maniac who Dr. Bruce Ivans worked in a biology lab, in a microbiology lab, so he had access to the anthrax, and he was just sending around politicians because he was a nihilist who hated everybody, and he just wanted to stir up more trouble by blaming it on Muslim extremists, right? So that there's all kinds of things like that were going on, but there were governments that actually looked into methods of using anthrax as a biological weapon, and they concluded that it's not a very good biological weapon because you have to expose somebody to a very concentrated burst of anthrax suddenly so that they inhale lots of spores. And basically, the only way you can do that is this way, by sending people letters. And in spite of that, this crazy man didn't even end up killing a single politician. He just killed a bunch of their secretaries that opened their mail. Right, so that's yet another layer of tragedy added onto that. Okay, Bacillus cereus. This topic is a little bit more fun because it's not as deadly as anthrax. Right, so it again produces endospores, but it is a common cause of food poisoning, particularly reheated steamed rice. Right, so if any of you are interested in a career in microbiology, I mentioned at the beginning of the lecture, at the beginning of the course, that one of the things you can do is you can become a public health inspector, somebody who inspects restaurants. Restaurant inspectors and food inspectors have to know about the common causes of food poisoning and so on. And Bacillus cereus, it contains the word cereal, right? So cereal, cereus, because this is a common contaminant of cereals, grains, and in particular rice, right? So if you have rice, it's possible that there is, in fact, Bacillus cereus endospores in the rice, right? That could easily happen. And if they germinate, they'll start to grow in the rice, and they produce, Bacillus cereus produces two exotoxins, one of them that causes diarrhea, and one of them that causes vomiting. Right? Now, the diarrhea toxin is what's called heat labile. I'll show you how to spell that in a minute. It's called heat labile, meaning that heat will destroy it, right? So if you have some Bacillus cereus growing in some rice, if you steam the rice or if you fry it in a wok or something like that, you will deactivate the diarrhea toxin because it is heat labile. But the vomiting toxin is heat stable, so heat will not kill it. Right? So if you have Bacillus cereus growing in rice and you steam it or you fry it in a wok and you eat it, you'll still end up vomiting, but at least you won't get diarrhea. Okay, so here they are producing endospores rather, and you can see the Bacillus cereus is just like Bacillus anthracis is a streptobacillus, right? So we would stain this. This has been stained with malachite green, and it hasn't been counterstained with saffron, and it's counterstained with something else. But anyway, here you see the bacteria. So the diarrhea toxin is heat labile. So you should know the word labile means that it can be deactivated by heat. So occasionally you get Bacillus cereus contaminating meat, but if you cook the meat, if you cook it, you won't get the toxin. You won't end up with diarrhea. However, one of the most common ways that people get food poisoning is if you have a restaurant that serves rice. And typically, a restaurant that serves rice will serve different dishes. Some of them involve steamed rice, and some of them involve fried rice. So what will happen is you'll have a restaurant where somebody will make up a batch of steamed rice, and then at the end of the day, they won't sell it. They'll say, okay, well, I've got half of this big container full of steamed rice that I couldn't sell today. So what I'll do is I will put it in the fridge, and then tomorrow I'll make fried rice out of it. And what you do is so it was, in fact, what happened was that it was sitting, if it was contaminated with Bacillus cereus, it was sitting at room temperature, or it was sitting in the kitchen of the restaurant all day for 12 hours or something like that, while the Bacillus cereus was growing and producing toxin. Both of those, diarrhea and vomiting toxin. So then the next day, the restaurant owner goes and fries it into fried rice, and the diarrhea toxin is deactivated, but the vomiting toxin is still there. So then a whole bunch of people that eat the fried rice that day will end up vomiting, right? So that's a common mistake that restaurants make, and so as a health inspector, you would have to check that and make sure that they're not doing that. Any food that's not consumed is supposed to be thrown out at the end of the day. In fact, even depending on what type of food it is, you may have to throw different types of food out several times. Right? So one thing you may not know is that, that if you poison your customers with food poisoning, you can be sued. And large companies realize this because large companies have lawyers. Little restaurants generally don't have lawyers that advise them about this. So large companies know this, and so McDonald's is one of the largest companies in the world, and they throw out their hamburgers every 20 minutes. In fact, these days they don't even cook any hamburgers unless you order them. But when I was young, a friend of mine had a job in McDonald's, and he was telling me that the McDonald's used to cook hamburgers, and in the early days they would cook hamburgers and they would leave them under an infrared light to keep warm until somebody bought them. But if nobody bought them in 20 minutes, they would have to throw them out, and so that's why probably a few years later they discovered that it's more profitable to simply cook them on demand than to throw them out every 20 minutes. All right, just a little joke. Reheated rice for dinner, be serious. All right, now let's look at two cocci, important cocci. So within the phylum Firmicutes, we still have the low GC content gram-positive, and here up at the top is our old friend Staphylococcus, genus Staphylococcus, and then below it we have genus Streptococcus. Right, okay, so example, Staphylococcus aureus, which is part of the skin microbiota, and it's an opportunistic pathogen. And Streptococcus pyogenes is some groups, depending on the type, some of them are hemolytic, and they can usually be identified using the Lancefield grouping, which we'll talk about later. Starting with Staph aureus. Right, a few words about Staph aureus. Okay, it produces the glycohydrolases that destroy hyaluronic acid, and an alpha toxin as well. And under the right circumstances, Staphylococcus aureus can cause all of these problems over here on the right. Right, food poisoning, acute bacterial endocarditis, osteomyelitis, and you should memorize these diseases well enough to recognize them on an MCQ question as well. Okay, so this is what it looks like. Of course, it is a Staphylococcus. It's a gram-positive Staphylococcus. It produces 7 heat stable exotoxins. So you can, if you have staphylococcal food poisoning, staphylococcal contamination of food, cooking it will not get rid of it. It also has 5 cytolytic toxins. It produces a toxin called toxic shock syndrome toxin 1, TSST. That one is important because there's a historical anecdote about that. TSST1, toxic shock syndrome toxin 1. It produces a protein called protein A as well. And then it is also the cause of an antibiotic. There's an antibiotic resistant version of it called methicillin resistant Staphylococcus aureus. That's where the term MRSA comes from, and that will become important later when we talk about antibiotic resistance. All right, now, food poisoning is a common problem. Staphylococcus aureus is a common contaminated food poisoning, particularly with custard products. So if you're interested in restaurant cooking and things like that, custard products are eggs and sugar. Eggs are mostly protein. Sugar, of course, is sucrose. Those are the two things that bacteria like to eat most. And so if you cook custard products like a custard pie that's shown here on the left, let's say the cook mixes up and gets some Staphylococcus aureus from his fingers into the batter, and then they cook this custard pie. Now, let's say 200 after cooking, 200 of the Staph aureus cells have survived, or Imagine somebody cutting a slice out of that pie with a dirty knife that had a little bit of Staphylococcus aureus on it. If you, it's a common thing for people to take pies and things like that to picnics at the beach or something like that. So I want to just imagine you started out at the beginning of a day with a custard pie at a picnic or something. You have a company picnic, and a few people eat a piece of the pie and they contaminate it with a knife, and so you get maybe 200 cells in the pie at 8 o'clock in the morning, and then it sits out in the hot sun at 37 degrees for 12 hours. How many cells would we have at the end of the day? Right. So remember that under optimal conditions, at 37 degrees, bacteria like E. coli and Staph aureus will divide every 20 minutes, right? So if you have 200 cells when you cut it with a knife at 8 a.m., 20 minutes later you'll have 400, 20 minutes later you'll have 800, then 1600, and so on. Right? And for the rest of the day, you'll actually end up having quite a lot of Staph aureus, and enough to actually cause food poisoning, even if it's not a virulent strain of Staph aureus. Acute bacterial endocarditis, which is where the bacteria actually gets into the body and starts growing around the valves of the heart. That's another thing, right? So if I asked you which of these bacteria causes acute bacterial endocarditis, you would answer Staph aureus. Osteomyelitis is bacteria growing in the joints of bones, right? That's another thing that some virulent strains of Staph aureus cause, and that's why Staph aureus is one of the more important bacteria. Impetigo is a skin disease. I should have warned you before showing you that, but it is where you get staphylococcal aureus infection of the hair follicles. Folliculitis is the same thing basically, except that it's a less severe version of impetigo. A scalded skin syndrome is where you have Staph aureus growing just under the top layer of skin, causing the top layer of skin to peel off, so it looks like you've had a bad, like you've been scalded with boiling water, or like you've had a sunburn, bad sunburn. Okay, I mentioned toxic shock syndrome toxin 1, right? This is a particularly powerful type of an antigen. An antigen is an agent that causes a severe immune reaction. TSST1 caused the deaths of a dozen women or so in the early 80s because tampons were contaminated. There was a batch of tampons produced by the Rely tampon company that were contaminated with a virulent strain of Staph aureus, and it actually cost several women their lives due to toxic shock syndrome. So there was a bad batch of Rely tampons that caused women to die from a anaphylactic shock, which they named toxic shock. They couldn't figure out what was causing it until a microbiologist tracked down the tampons. Epidemiologists basically saw that the only thing that all of these women that died had in common was that they were all using Rely tampons, and a particular batch of them. So that is where the condition that killed these women was referred to as toxic shock syndrome. Later on, they discovered that toxic shock syndrome was caused by TSST1 toxin, which is produced by Staph aureus. And so this company had produced a bad batch of tampons that were contaminated with Staph aureus. Occasionally, Staph aureus can get into the lungs. It tends to be localized to one lobe. Nobody knows why. So if it gets into the lungs, if you inhale some of it, it can cause pneumonia, and it can occasionally cause meningitis, which is inflammation of the brain membrane surrounding the brain. It sometimes is caused by an infected. Brain surgery is done on somebody, and the incision gets infected with Staph aureus, right? Because they weren't paying enough attention to keeping the dressings of the wound clean. Let's move on to Staphylococcus pyogenes. Streptococcus rather. It forms strips rather than bunches. Okay, so Streptococcus pyogenes, I'm going to talk about what the Lancefield grouping is in a minute, but it is a member of the Lancefield group A. So you need to memorize this. If I ever ask you, give me one example of a Lancefield group A bacteria, you can say Streptococcus pyogenes. Right? It is generally caused by droplet transmission. It has a capsule, so which makes it harder for the immune system to get rid of it. It produces an antigen called the Sp antigen, which again causes toxic shock. And more importantly, it produces a protein called the M protein. The M protein has a very similar structure to the muscles in your heart. So if you get an infection of Streptococcus pyogenes when you're a child and your heart is still developing, what will happen is that your immune system will make antibodies that are meant to attack the M protein, but because the structure of the M protein and the structure of your heart muscle are very similar, that antibody will cross-react with the developing muscles in your heart, causing your heart to be weakened. Right? So the disease that you would get as a child is called scarlet fever. Right? And before the invention of antibiotics, people who had scarlet fever as a child would end up having heart problems in middle age, and they would die of heart attacks because their heart had been damaged as a result of a Streptococcus pyogenes infection early in their life. So it was a heart damage secondary to a Streptococcus pyogenes infection. Okay, Streptococcus pharyngitis is where you have a streptococcal infection of a sore throat. Sorry, I should warn you that we are having disturbing images in this lecture. Okay, so streptococcal pharyngitis, the pharynx refers to the space behind your mouth, and so this is commonly called a strep throat, right? So if I ask you what is the cause of a strep throat, you would answer Streptococcus pyogenes. This is where, and Streptococcus pyogenes is often hemolytic, so the redness is caused by the fact that the bacteria is breaking or lysing some of the cells and releasing a little bit of blood from the tissue. Right, scarlet fever looks like this. One of the characteristics of scarlet fever is that your tongue turns bright red. If you're a mother and you see your child with a bright red tongue like this, take them immediately to the hospital so they can get antibiotics. And the antibiotics will kill off the Streptococcus pyogenes before it has a chance to produce the M protein and cause any damage to the child's heart. Rheumatic fever is where the Streptococcus pyogenes spreads to the whole body. Once again, if you have this rheumatic fever, you take them to the doctor immediately so they can get rid of the Streptococcus pyogenes before it can produce the M protein, thus indirectly causing damage to the child's heart from your own antibodies. So that's an autoimmune disease. As I said, if you have rheumatic fever as a child, then you end up having rheumatic heart disease, RHD, as an adult. It's a result of the antibodies that your own body produced attacking the heart muscle. Pericarditis is where strep pyogenes gets into the membrane surrounding the heart, causing irritation. All right, necrotizing fasciitis is caused by virulent strains of Streptococcus pyogenes. It's commonly known as flesh-eating disease or flesh-eating bacteria, right? And this is caused by the fact that you happen to have a virulent strain of Streptococcus pyogenes that is capable of making the hyaluronidase exoenzyme that causes it spread throughout the tissue, causing the tissue to break down, allowing the Streptococcus pyogenes to spread farther and then kill more tissue, eat the tissue. The tissue breaks down and the bacteria eats it, then it spreads more, kills more. So there are a lot of bacteria that decompose the animal body and eat it, but most of them have the courtesy of waiting until after we're dead before they do that. Virulent strains of Streptococcus pyogenes are impertinent enough that they start killing you and eating your tissues before you're even dead. So this again is an example of tissue that you have to, most of the time you can try to get rid of it with antibiotics, but if it doesn't work, you have to cut off the limb very quickly before it spreads too far. All right, let's take a little break here and give you a little bit of Canadian history and look at the intersection between Canadian history and microbiology. All right, so if you're interested in Canadian history, Here on the left, we have a Canadian politician named Lucien Bouchard. Right? He's from Quebec. Remember that Quebec is the Canadian province that has where they speak French. For many years, Quebec has been experimenting, or many Quebecois have had the idea that they want to separate from Canada and have their own country because they have a different language and a different origin and a different history than the rest of Canada. And so Quebec separatism is not as common now as it was 20 or 30 years ago. It probably hit its zenith, had its high point in the 80s, in the 1980s to the 1990s. And now this, do you know who this is in the middle? And so that's Brian Mulroney, who was the president. He was the Prime Minister of Canada in the 1980s. Right? Now, Quebec separatism was strong in the 1980s and the early 1990s, and the different Canadian politicians have tried to address the issue. The fact that at the time roughly half the people in Quebec wanted to separate and have their own country. Is there anything that the rest of Canada can do to make them happier to stay and whatnot? And so Lucien Bouchard was originally one of the solutions that some of the Canadian politicians came up with was something called asymmetrical federalism, where you give Quebec a few special powers that the rest of Canada doesn't have, and maybe that'll be enough that they'll decide to stay. And so asymmetrical federalism, also known sometimes as sovereignty association. Sovereignty association meaning that Quebec has limited independence within Canada. So that's another possibility. So Lucien Bouchard was a Quebec politician who was in favor of sovereignty association. And so Brian Mulroney was a federalist, of course, who wanted to have Canada be all together. And so he made a deal with Lucien Bouchard and said, if you support me, I'll make you a minister in my government, and we will address the issue of sovereignty association. We'll have a referendum across Canada, and it was called the Meech Lake Accord. And so we had this referendum in the 80s. And Lucien Bouchard said, "Okay, I'll try and convince Quebecois people in Quebec not to separate if we have this referendum and give Quebec a few extra little powers here and there." And so it was a gamble, right? And so we had the referendum. The referendum failed, and Lucien Bouchard. So the rest of Canada didn't want to give Quebec any special powers. Lucien Bouchard and a lot of other people who were in favor of sovereignty association became outraged because they weren't really asking for very much, in my opinion. But still, the rest of Canada said no. And so a lot of the people who were Quebec politicians who were in favor of sovereignty association were outraged and insulted, and they said, now we're totally in favor of separation, 100% separation. So Lucien Bouchard overnight went from being the the savior of the Canadian Confederation to the biggest separatist, right? So a lot of politicians and people in the rest of Canada thought of him as a villain. Right, so he was the founder of the Bloc Québécois, which is the federal party that wants to take Quebec out of Canada. Right, so there he is, gone from being a federalist to a separatist, banging the drum of Quebec separation. So here's political cartoonists making a lot of cartoons about Lucien Bouchard. Okay, so people who wanted Canada to stay together suddenly hated this guy. They thought he was a big villain, and then suddenly he develops necrotizing fasciitis. He develops a virulent form of streptococcus, and he had to have his leg amputated. Right, and so he is the most famous Canadian to have necrotizing fasciitis, and of course all the people that hated him before had to say nice things about him for a while, and then when he got better and walked with a cane and had an artificial leg, then they could go back to hating him again and saying terrible things again, which they did. And so one of the things was, I remember seeing one of the cartoonists interviewed on television saying he wondered how long it would be before he was allowed to start drawing nasty political cartoons about Lucien Bouchard again. And it didn't take long, I can tell you. So he walked with a cane suddenly, and all the comedians were wondering how long can we take before we make fun of that. So here's a political cartoon that resulted from that. This is when another Canadian Prime Minister named Jean Chrétien passed a bill called the Clarity Bill, which made it more difficult for Quebec to separate. And so here you see Lucien Bouchard's cane, the end of his cane, about to slip on a banana peel, which is Jean Chrétien's Clarity Bill. Right, so that's just a little Canadian history for people who are interested in Canadian history. This is an intersection between microbiology and Canadian history. So Lucien Bouchard is the most famous Canadian ever to get necrotizing fasciitis from strep pyogenes. I will not ask you that on a test. I will never ask you that on a test. That was purely for entertainment purposes. Okay, so the Lancefield groupings is a method of categorizing bacteria, particularly pathogenic bacteria, based on you have a panel of antibodies. You have a bunch of different antibodies, and you take a little sample of the bacteria and you mix it with each one of these antibodies, and then you see which antibodies the bacteria will react to. And then you can put the bacteria into groupings and figure out which bacteria is infecting somebody based on the reaction to the Lancefield antibodies. Okay, so let's take a little detour and discuss the Lancefield grouping system. It's a method of grouping and identifying bacteria that is based on which antibodies the bacteria will react with. You do need to know what the Lancefield grouping system is and how it works, and you do need to know that it is a way of classifying and discovering when somebody's infected with a bacteria. You want to discover which bacteria it is. This is a method of doing it. Okay, so this is named after a famous female scientist named Rebecca Lancefield. She's probably the most famous female microbiologist ever. So if you're interested in the history of women in science or feminism, or just generally the history of women in science or the history of microbiology, then you should know who Rebecca Lancefield is. I will not ask you her name on a test. I will not, but just you should know that if you're interested in the history and the influence of women in science. So Rebecca Lancefield invented the Lancefield grouping system. Notice she's sitting here with all of these plates, all of these Petri plates, McConkey plates and blood agar plates and so on. Okay, so this is another way to identify bacteria based on you mix bacteria with water, and then you divide up these bacteria that's mixed with water, and you add some antibodies, and then you look to see which bacteria will agglutinate. Agglutination means to clump together, right? So you mix the bacteria with antibodies on a special card. You actually buy the Lancefield grouping kit. So they send you a little box with some little droppers, bottles filled with droppers, and then each of those bottles contains a different antibody. And so you mix each of those antibodies with the bacteria to see which one causes the bacteria to form clumps or to agglutinate, which antibodies cause the bacteria to agglutinate. And then you put the bacteria into groups, and the groups are labeled A through S, right? So remember this, you must know this, that Strep pyogenes is a Lancefield group A, is a group A Lancefield microbe. So here's the little kit that you get if you buy the Lancefield kit. Those bottles in the background there contain the different antibodies. They give you a little card that has little wells in it. So you mix the bacteria with water, you add a little bit of it to each of these little circles, and then in turn you add these antibodies to each of the little circles. And so this area up here, what you see there is agglutination. What you see here is non-agglutination. So based on the fact that this bacteria reacted with that antibody but did not react with that antibody, you can use it to classify it as a Lancefield group A, for example. Right, so two things you have to remember. Number one, Lancefield group A includes Streptococcus pyogenes. So if I ask you for an example of a bacteria that is a member of Lancefield group A, you say Streptococcus pyogenes. The enterics, most of the enterics are members of Lancefield's groups D and E, so I might ask you that as well, right? So I can show you 5 bacteria, only one of which is an enteric, and then I'd say which one of these is a member of Lancefield group D, or most likely to be a member of Lancefield group E. All right, quickly, and then we're finished with this weekly dose of pain. The Archaebacteria, these are extremophiles that are left over from the time when the world was very hot and had a lot of methane and sulfur in the atmosphere. The world is not like that anymore, thank goodness, otherwise we wouldn't be here, but those bacteria are still around. And they live in extreme environments that still exist in the world. Okay, two examples. The phylum Crenarchaeata, these are cocci that are hyperthermophiles. So they live in ambient temperatures of around 100 degrees, and they oxidize sulfur and convert it to sulfuric acid. So where do you find these things? You find them growing near volcanic vents in the ocean or in hot springs, right? So if you go to the Banff Hot Springs, which is in Alberta, it's a famous tourist resort. There's an underground volcano. Water is coming out of the underground volcano, so you get this hot water that's coming out, and people bathe in it. And the water is filled with sulfur, and you can smell sulfur. And that water has a bit of Crenarchaeata bacteria in it because that's where they like to live. Right? And then the other group, the Euryarchata are the methanogens. So methanogens, gens means to produce or to cause, and then methane refers to methane gas. So what they do is they actually consume carbon dioxide, which is kind of useful if we're worried about global warming, so biotechnology. And they consume hydrogen. That's neither here nor there. But they produce methane gas, right? So one neat biotechnology application that you can think about for the future, if any of you become biotechnologists, is that you can take these and you can put them into, you can make a water sump for a smokestack in a factory so that you have all these fossil fuel emissions, carbon emissions bubbling through a tank that contains these, and they will actually soak up the carbon dioxide and they'll produce methane, which you can then use as another fuel. So you can use them because they produce methane. You can convert the methane into methanol, and then you put the methanol into regular gasoline, and then you make biodiesel, what's known as biodiesel. Right, so here we have the Crenarchaeata living near hot springs and volcanic vents. That's a hot spring on the left. That's a volcanic vent at the bottom of the ocean on the right. The temperature near that volcanic vent is around 100 degrees. It's hard to believe that things can live at that temperature, but lots of things do. In fact, these are things that are left over from 2 billion years ago, and they're still going strong in some of these areas. And the methanogens, the Euryarchaeata, can be used to turn sewage into methane, and the methane can be used to generate energy. All right, so we talked about quite a few things in this lecture. We talked about the types of symbionts. We talked about the different types. We talked about obligate intracellular parasites like Rickettsia, which also happens to be pleomorphic, and so on. We learned about the important gram-negative phyla, particularly the proteobacteria, and then we have alpha, beta, gamma proteobacteria, and so on. We learned about the important ones. We learned about the Firmicutes and the Actinobacteria, which are gram-positive with either high or low GC contents in their genomes. We learned about the enterics. We learned about the Lancefield grouping system after Rebecca Lancefield. And we learned two examples of Archaebacteria. Okay, so the alpha protobacteria, the important ones, we learned about these and their characteristics. The betas, we learned about those. The gammas, we learned about those. The epsilons, we learned about those. Those are the proteobacteria. Then we learned a couple of spirochetes and planctomycetes, which are also gram-negative, but are not members of the proteobacteria phylum. We learned about the gram-positive actinobacteria, these ones. Frankia californiensis is used for nitrogen fixation by legumes. Right? We learned about the Firmicutes. Most of them are bad. Right? Lactobacillus acidophilus is an exception because it excludes Candida albicans from the human vagina, for instance, and we also use it to ferment different dairy products like yogurt. All right, so that's it for this one. I believe our next lecture, the next lecture I believe is on the important protists.